Good morning, everybody. First of all, thank you for giving me the opportunity to present the results here. And today I will discuss about how biomolecules fall from a random coil configuration to its native functional structure. The first folding process that I will discuss about is the DNA hybridization. This process is the process where two independent single-stranded DNA met each other, forming the double helix that was discovered by Watson and Crick. And in this process, the hydrogen bonds and the stacking interactions of the base pairs takes place. It is also important to note that from bulk experiments have demonstrated that this process presents a heat capacity change different to zero. In particular, depending on the length and sequence of the molecules studied, the value of this heat capacity change range from 40 to 160 calories Kelvin mole per base pair. The second process that I will discuss about is how proteins fall. Since 1961, when Christian Anfinsen claimed that the native structure of small globular proteins depend only on its amino acid sequence, this topic became a hot topic because, as Cyrus Levinthal noted, according to the Anfinsen dogma, proteins will require astronomical timescales to fall due to the large number of possibilities. However, the experiments at the moment demonstrated that proteins fall in the second and millisecond timescales. So, according to this experimental evidence, Levinthal proposed that protein folding must be guided by the rapid formation of local interactions to speed up this process. Two of the most accepted hypotheses that explain how proteins fall are the molten globule and the energy landscape hypothesis. The molten globule hypothesis was proposed in the late 80s by Oleg Pitsin, claiming that an intermediate state similar to the native state, but with most of the native interaction not yet stabilized, is necessary to form the native structure. The energy landscape hypothesis is an hypothesis claimed by Peter Wollines and co-workers based on the experimental evidences that small globular proteins can fall in a two-state manner. In this hypothesis, the energy landscape that governs the protein folding is a free energy landscape with a funnel shape. I will test this hypothesis using a two-state manner, a small globular protein named Baronase, but also I will check if this energy landscape hypothesis explains how DNA hairpins form the double helix plus loop structure. To do so, I use single molecular experiments using an optical trap instrument. The optical trap is based on the original idea proposed by Arthur Ashkin, where two counterpropagating lasers are focused in the focal point by suppress the scattering force and enhance the gradient force to trap small dielectric objects. This instrument has an extra collimated laser that can raise the temperature surrounding the optical trap to change the temperature that the molecules are feeling. To do experiments with this setup, it's necessary to tether the molecule that you want to study between two dielectric objects via double-stranded DNA handles. One bit or end will be controlled using the optical trap, while the other will be kept fixed at the tip of a glass micropipette by our suction. By varying the position of the optical trap relative to the micropipette, we will exert different force values to the molecule. In particular, at a given force and time, the molecule can be found in the native state or in the unfolded state. Two different experiments are commonly done using this setup. The first one is the hopping experiments, where the optical trap is kept fixed at different positions, while the force signal is recorded as a function of time. For a two-state hairpin, as you can see in this figure, we can clearly distinguish two force levels, one when the molecule is folded and the other when the molecule is unfolded. The other kind of experiments that are commonly done are known as pulling experiments. In these experiments, the force is run from a minimum force value, where the molecule is always in the native state, to a maximum force value, where the molecule is always in the unfolded state. The unfolding and folding events are shown as sudden force rifts in the force distance trajectories, as we see here. The force that each state feels presents in the force distance curve and also in the hopping experiments depend on the elastic properties of the system. Commonly, the optical trap is modeled using the Hooke's law with a constant spring. On the other hand, the folded hairpin or protein is modeled using the semi-flexible elastic model known as free Legendre chain, assuming that the Coulomb length equals the distance, the thickness of the double-stranded DNA, and the MC termini distance, that is the distance where we exert the force in the proteins. On the other hand, the unfolded molecules and also the double-stranded DNA handles are modeled using the Worldline chain model that characterize the elastic response of semi-flexible polymers using a persistent length and an interphosphate or interamino acid distance. First of all, I studied the DNA folding using these hairpins that you see here. On the one hand, we can clearly see 3 hairpins, short hairpins with different GC content. These are the poly-C, poly-A and mixed hairpins, that I will use them to study if this heat capacity change that I mentioned before presents a sequence effect. And the other hairpins that has the same stem, I varied the loop length to study which is the contribution upon bending this loop when we need to form a structure. This will be analogous, for example, from these random parts of the proteins that are after or before a beta sheet or an alpha helix. To study this, I carried out 2 kinds of experiments. For the different GC content hairpins, I carried out hopping experiments at different temperatures, and to study the lab contribution, I did pooling experiments at different temperatures. A general aspect we can see here is that when we reduce the temperature, the force necessary to unzip the molecules goes up. This makes sense because, as we can imagine, if we increase the energy in the surrounding media, we need less energy to open the base pairs. Also, we can note that in the unfolding and folding trajectories for the loop 8, loop A adenines and loop 20 thymines, we can clearly see an hysteresis between the unfolding and folding trajectories. Due to this hysteresis, I couldn't do hopping experiments with these hairpins. From this data, I extracted the kinetic rates for the unfolding kinetic rates for these molecules. I derived the kinetic rates from the average. Yeah.
So in the previous graph you showed us, in the folding experiments, they are in some cases here.
Yeah. Okay. You mean between molecules or between temperatures?
I guess I'm thinking about it between the folded and unfolded.
Okay, so for example, in the loop 4, it looks parallel, while in the other ones, no. This comes from the fact that the elastic properties of DNA are really strong force dependent. So, for example, for the small loop, as I'm opening and closing in a narrow force range, we cannot see this effect. But when I pull big loops, then large unfolded molecules compared with the folded ones, I clearly distinguish this difference in force range. So, as I said, I determined the kinetic rates from this data. For the hopping, I measured the average, the inverse of the average resident time. And I treated the unfolding trajectories as Markovian processes to determine the kinetics from the distribution of the first form that changes its state. The kinetic rates are shown here. And if we note, when we study the sequence effect, we can clearly see that the kinetic rates for the poly-GC takes place at larger forces than the kinetics for the poly-AT and mixed hairpin. It makes sense because, as I mentioned before, the energy to open a GC base pair, well, as we know from the Watson-Crick and other studies, the energy to form a GC base pair is higher than the energy to form an AT base pair, and this is supported with these experiments. Relative to the loop effects, we can clearly see different things depending on what we see. If we look the unfolding trajectories, this is the left panel, we can clearly see that the symbols collapse, but the colors no. It means that the unfolding kinetic rates do not depend on the loop size, but depend on the temperature. In contrast, the folding kinetic rates, we can clearly see that the symbols collapse, but the colors collapse, but the symbols no. It means that the folding kinetic rates depend on the loop size. This is indicative of a folding process entropically driven. I would also like to note that the squares and the triangles that are the 20 basis loops depend on the sequence of the loop. we can see that the loop for the 20 thymines bases is faster than the 20 adenine bases loop. This is indicative that the energy that you have to stack the bases in the loop is affecting the kinetics. I will discuss this a little bit later. To determine the free energy landscape from this data, I determine the total free energy difference between the native and unfolded state. I determine the entropy difference between the folded and unfolded state, and also the enthalpy difference using these equations. The first one is the detailed balance conditions that relates the unfolding and folding kinetic rates with the folding free energy and the stretching contributions to stretch the unfolded and orient the folded molecule. To determine the entropy, I use a Clausius-like equation, and to determine the enthalpy, I use the Gibbs free energy definition, taking into account that we know the energy and entropy from the previous equation. First of all, I determined the folding free energy with a novel protocol that I found that is general and can be used for different molecules, and I will discuss a little bit now. The idea is, using trial values of delta G0, we reconstruct the unfolding and folding kinetic rates at the force where the unfolding and folding events take place. These are the anti-symbols. Varying this value, we can see different regimes, for example, the 2, 10, and 18 kilocal moles. Then, by fitting the unfolding rates to this parabolic function and the folding ones to the equation that we know from the detailed balance condition, we calculated the chi square relative to the fits to the experimental and reconstructed values. The value that minimizes this chi square is the value that we consider as the folding free energy for a given temperature. Using these reconstructed kinetic rates, we estimated the existence force to estimate the enthalpy and entropy values. The values are shown here. Relative to the energy, we can clearly see that the loops with only adenine bases more or less collapse, suggesting that the energy to bend the loop is negligible, while the loop formed by only 20 thymines is slightly less energy cost than the others. This suggests that the 20 adenine bases are more stacked than the 20 thymine bases, which makes sense because we know from the literature that the pyrimidine motif are more strongly stacked than the pyrimidine motifs. Relative to the entropy and enthalpy difference, we can clearly see that we have a different slope depending on the loop. It suggests that the loop affects the total heat capacity change that we have. I will discuss a little bit this value later. Relative to the sequence effect, we can clearly see that, as I mentioned before, the energy to unzip or to form a GC base pair is larger compared to the AT base pair. These values are basically the total energy that we measure for each hairpin divided by the number of bases or base pairs in the stem. The dashed line that we can see in these plots is the expected value considering the results for the poly GC and poly AT hairpins compared with the mixed hairpin. The difference comes from the fact that we have some purine motif in the stem of the mixed hairpin, and we hypothesize that the difference will be equal to the difference that we have seen in the 20 adenines and 20 thymines loop. I will discuss later. Also, we can see that the slope in the entropy and enthalpy values is different depending on the GC content, suggesting that effectively we have a heat capacity change that depends on the sequence or the kind of base pair that we are forming. However, in all these results, we have the contribution to bend the loop and stabilize the double-stranded strand. So, to decompose these two contributions, we have characterized the transition state between the native and unfolded state. To do so, we have fitted the reconstructed kinetic rates at zero force to these equations that are basically an Arrhenius equation decomposing the energy in terms of entropy and enthalpy. The results that we obtain are summarized in this table, and I will discuss different parts of this table that give us the idea of how these molecules fall. On the one hand, we can see that the enthalpy energy, the enthalpy difference between the native and the transition state, this is the dagger, is larger compared with the same difference between the transition state and the unfolded. This is the star. This is indicative that in the collapse from the transition state and the native state, most of the van der Waals weak interaction and hydrogen bonds take place. In contrast, the heat capacity change between the native and the transition state is much smaller than the heat capacity change between the transition state and the unfolded state. This is indicative that most of the configurational changes take place between the unfolded and the transition state. Based on this evidence, we suggest that at the transition state, the two single-stranded strands are aligned and facing each other to facilitate the collapse and the formation of the double helix. The results are supported by previous fluorescent single molecular experiments that studied the elastic properties of two independent single-stranded chains when they meet each other before stabilizing the double helix. Finally, I have compared the difference in energy per purine motif, comparing the different 20 bases loop and the difference between the poly-GC and poly-AT results and the mixed one, finding that the difference in energy per purine motif equals 0.3 kilocal mole, which is in agreement with previous experiments based on molecular dynamics and single molecule experiments. To conclude, I did the same kind of experiments using a protein that is Barnes, as I mentioned before, that falls in a two-state manner and has been used to determine the phi alpha analysis. That it's an analysis that allows us to characterize the transition state properties. To do so, I did pooling experiments because, as you can see here, proteins tend to be a highly dissipative molecule. And you can see here that the folding events are these really small jumps and occur at really low forces. From the first rupture force distribution, derived the kinetic rates that I did before, and reconstructed the kinetic rates from 0 to 30 piconewton using the detailed balance condition, as I did before. Using the energy, enthalpy, and entropy values that we derive from protein varnish are shown here, and we can clearly see that the entropy and enthalpy values present a temperature dependence. The heat capacity change that we measured for this protein is around 1,000 calories Kelvin mole, which means that the protein folding changes around 1 calorie Kelvin mole per degree of freedom that we lost during the collapse from the random coil to the native state. Finally, I characterized the transition state properties to see if we can discover if there is a molten globule state for this two-state protein. And what we found is that the thermodynamic properties of the transition state are the following. The heat capacity change between the native and the transition state is roughly 9 times smaller than the heat capacity change between the transition state and the unfolded state. This suggests that at the transition state, the protein is more or less well organized, but the hydrogen bonds are not stabilized. Also, this is supported by the fact that the enthalpy and entropy difference between the native and transition state are 10 times larger than the difference between the transition state and the unfolded state. So, in global, we are seeing that the transition state has the thermodynamic properties of this molten globule state proposed by Oleg Pitsin in the late 80s. Also, we can see that the free energy landscape governing this folding and also the folding for the DNA hairpins looks like a golf course, where the plate would be in a space where different configuration can be placed. So basically, we are triggering with the amino acids or bases, but when we arrive to the hole, it would be the transition state, all the native contacts form and falls to the ground of the hole of this golf course. So this idea is summarized with these three strong temperature inequalities, and this is general for DNA and proteins. To conclude my talk, I would like to remark that we are doing now experiments with a protein that presents an intermediate state. In fact, it presents two intermediate states, and we want to characterize the thermodynamic properties of this intermediate state, but also the transition states for this intermediate state to see if we can see some general feature for this transition state. And we will try to connect this energy landscape hypothesis with the novel hypothesis of the fold-on hypothesis, where it's claimed that the protein falls in different steps by forming intermediate states, but independently each other or of the others. Finally, I am also finishing an analysis using long DNA hairpins in order to study the enthalpy, entropy, heat capacity change, and folding energy for the 16 DNA motifs that form the nearest neighborhood model, finding that, in fact, we can clearly see a difference in energy, enthalpy, and enthalpy, entropy, and energy for the different 16 motifs, as expected for the literature, but also we are finding a strong sequence effect in the heat capacity change. It would be interesting because this can be used to improve the PCR experiments where the temperature is really a trick element to improve. To conclude, I would like to thank Felix Ritor to giving me the opportunity to discover what is the research, but also I would like to thank Jordi Alcaraz, who gave me the opportunity to change from the single molecule world to the cells, now studying lung cancer and metastasis. So thank you for your attendance.
DNA folding the transition state would be partially folded by a certain number of days.
Okay, yeah. So you mean, for example, that half of the stem is, yeah. If you think in that way, the transition state would be a minimum in the free energy landscape because you have some part that is folded. So if it is a minimum, it's not a real transition state, would be an intermediate state. But also what we are seeing is that one thing is what we commonly drive as free energy landscape, that we put the number of bases and an energy profile. So commonly from 0 to 20, for example, you find a maximum in the 10 base pairs in the 10, so in the middle. The problem is that what we found is that when we carry out these experiments, we found that the energy is in agreement in the fact that 10 base pairs are open, but not the enthalpy and the entropy that were not analyzed with previous experiments because this idea of using an extra collimated laser to change the temperature was not possible. Also, in this work that I mentioned by Shembla, where he studied independent single-stranded DNA hybridizing, they found that at the transition state, any contact was made. So was only organized the single-stranded DNA chains facing each other and adapting the interphosphate distance to help the collapse. So this means that probably you know the interphosphate distance for the single-stranded DNA is around 0.6 nanometers, but the interphosphate distance for the double-stranded DNA is the 0.44 nanometers. So basically what we need is to compress the single-stranded DNA and align to form the hydrogen bonds. So not hydrogen bonds is formed before. But energetically speaking, could be understood as part of the hairpin is formed. But in fact, what we are seeing now is that not. Or maybe yes, but one base that is in our resolution, experimental resolution. I think yes, because I think Matteo Capitano, I don't remember the name, but Capitano is the last name, is doing experiment with single molecule optical tweezers experiments, making bilayers, tissue, yeah, I will say tissue, but not tissue, frame of bilayer proteins. And he is doing experiments to characterize the thermodynamic and elastic properties of these organized cells, eye cells, proteins. But I don't know what he can do or not. Because in my experiment, if I pull in this direction and the proteins are organized in that way, I will break the bilayer organization. But maybe turning 90 degrees, I will modulate the organization, but I don't know how to do it.
I'm just curious, like, the main
Yeah. Well, basically, when you have these kind of experiments, you can do the fluctuation theorem that relates the energy to open with the energy to close. Or you can do what I did, that it's basically classifying events depending if you are opening or you are closed. What the problem with the with the methodology of the fluctuation theorem is that you need to subtract the stretching contributions, and these contributions depend on the elastic properties. And if you don't know how these elastic properties change in temperature, you are a little bit in conflicts. But in our case, we studied these elastic properties that are in this reference, the second one here, and we found that the elastic properties of DNA, but also protein, Barnes, change linearly with temperature. It's pretty easy to control this contribution.
Okay, let's thank Marc again.
Perfect. Hello, everyone. Before I start, I would like to thank the organizers of this symposium for giving me this opportunity to present my research here. My name is Jules Kenyi. I am a fifth-year PhD student at the University of Washington, and I work with Professor Marc. Today, I want to talk to you about how I've been using the computer simulation to study the dynamics of this protein called calmodulin and its interaction with the targets. Specifically, I want to show you how we use the combined experiment and simulation to show that as the target binds to calmodulin, it tunes its calcium properties. So here is the timeline of my presentation. I will first give you the reason why I'm interested in this topic, and then I will present the calcium calmodulin and the peptide that I'm using in this study, which is the calcium calmodulin dependent protein kinase 2, CAM-kinase 2. I will then present our coarse grain method and the result from the simulations, and I will conclude this work. So the calmodulin is a small molecule of about 148 amino acids, which is found in all eukaryotic cells. Upon calcium binding, the calmodulin undergoes multiple conformations, which allow it to bind and modulate the activities of over 300 target proteins, and including the calcium calmodulin dependent protein kinase 2, CAM-kinase 2. The CAM-kinase 2 is a large molecule which is involved in many cellular functions, including memory and learning. And as the calcium binds to the calmodulin, actually, its affinity for the CAM-kinase 2 increases. At the same time, as the CAM-kinase 2 interacts with calmodulin, the calmodulin affinity for the calcium also increases. So this complex mechanism between calcium, calmodulin, and CAM-kinase 2 is at the origin of many biological functions, including memory and learning, as I said. Due to the large size of CAM-kinase 2, only the calmodulin binding domain of CAM-kinase 2 is often used to study its binding properties with the calmodulin. Here, I'm showing you the resolved structure of the complex between the calmodulin and the binding domain of the calmodulin, from the CAM-kinase 2. You can see that the calmodulin wraps around the peptide and forms this compact structure. At the same time, the peptide adopts this helical conformation. However, it has been shown that in solution, this peptide actually, has a disorder structure, as you can see here. At the encounter between the calmodulin and the peptide, both undergo multiple conformations, which allow the calmodulin to wrap around the peptide and the peptide to adopt this helical conformation. In order to have a better understanding of this peptide in solution, a combined experiment based on circular dark Rosen and the all atomistic simulations were performed on a white type peptide known here as RRK, and it's 1 amino acid mutation, RAK, and 3 amino acid mutation, AAA. The experimental data show the presence of a secondary structure in a peptide with triple mutation, which does not exist in a peptide with, with one amino acid mutation, or even in a white type peptide. Since RRK are the charge residues, this implies that each mutation in the charge residues actually reduces the disorder content of the peptide structure ensemble. This has been confirmed using the all atomistic simulations, where I'm showing here the 10 most populated conformations for each peptide. As you can see, the peptide with triple mutation is dominated by this beta hairpin structure, as compared to the peptide to the white type peptide, which is dominated by the by the disorder structure. From these observations, we then ask the following questions: how do these peptides bind to calmodulin and affect its calcium affinity? And also, we want to know how do these mutated peptides interact with calcium calmodulin? In order to answer these questions, we conducted the experiment based on stuff flow measurement, where we measure the calcium dissociation rate from the calmodulin as a function of time. As you can see, calcium quickly dissociates from the calmodulin in the presence of the peptide with triple mutation, where the dissociation rate is almost similar to that of the calmodulin free target, as compared to the calmodulin in complex with the white type, or the calmodulin in complex with RAK, which is the peptide with 1 amino acid mutation. So I do want to point out that these observations happen mostly in the C domain of calmodulin, since in the N domain of calmodulin, the change, we did not observe any considerable change. And then, since the mutated peptides, which are RRK, are actually the charge residues, we then speculated that the electrostatic interaction between these charge residues and the acidic pockets of the calmodulin may play a crucial role in strengthening the calcium binding site of the calmodulin. In order to, yes. Okay, so here we have the calmodulin in complex. So this one, the green one is the calmodulin in complex with the with the peptide with triple mutation. So if you look at the time it takes for the calcium for all the calcium to be released from CAM, you can see that it's actually a short time, as compared to the calmodulin in complex. Yes.
You described the rate of calcium release from the N terminus.
Yes. So our collaborator did that stuff flow experiment where they measure the calcium dissociation rate from either first of all from the C domain of CAM alone, without they isolated the C domain of CAM, then they measure the calcium dissociation rate, and then now they do the same with the whole length CAM. Does that answer your question? Yeah. So, okay. So from this, then we want to use the coarse-grained molecular simulations to test this speculation. There are many tools out there to conduct the MD simulations. However, due to the importance of the calcium in tuning the dynamics of calmodulin, you can see that most of those tools lack this ability to capture the calcium to capture the calcium effect. In order to overcome this difficulty, we then use the coordination chemistry of calcium, which has been shown to adopt the pentagonal pyramidal geometry, as you can see here. And then we assign the calcium charges to the negatively charged residues present in that coordination chemistry, as you can see in this in the table. So then we added all this setup to the protein force field called OSM. OSM stands for associative memory, water-mediated structure and energy. So OSM uses a tree-based system to represent each amino acid, as you can see here. And the Hamiltonian used in OSM is given as follows, which is composed mainly of two terms. So we have the physics-based term, which is common to all the protein, and then now we have the bioinformatics-based term, where the equation is given as follows. So I will come back to this on my next slide. So the complex calcium, calmodulin, and CAM-KANase2 peptide has multiple minima due to the fact that they sample multiple conformations in solution. So you can see that. Also the energy barrier between those so the energy barrier between the conformations is actually very large. So you can see that it may require longer simulation time in order to sample the whole configurational space. In order to avoid that, we use the enhanced sampling based on umbrella sampling, and we further use the multi-state Bernard acceptance ratio in order to group the data for further analysis. So on my previous slide, so I told you that the OSM force field uses a tree-based system to represent each amino acid. Then now you can see that important details may be lost during that coarse graining. This is exactly where the bioinformatics terms comes in. It makes use of the known information from the. It makes use of the information from a known structure, not only to improve the configurational search of the global minimum, but also to record the important details that were lost during the coarse graining. And in this work, for the calmodulin, we use the known structure from NMR and X-ray crystallography. And for the calcium binding loops, we use the known structures from the first principle. From the first principle. And for the peptide, for the binding peptide, we use the known structure from the all atomistic simulations. Where I'm showing here the 10 most populated structures from used in our memory file. Here is an example of our, so here's an example of the calcium dynamic from our simulation. As you can see, the calcium calmodulin actually samples multiple conformations back and forth due to this high flexibility of the central linker. And in the presence of the peptide, you can see that the calmodulin wraps around the peptide to form this compact structure. And this peptide adopts the helical conformation. Then now, in order to better understand how this peptide actually interacts with the calcium, with the calmodulin, we then plotted the 1Z free energy profile of each complex as a function of the radius of gyration of calmodulin. I do want to remind here that the radius of the RG of calmodulin has been determined either experimentally or numerically to be around 22 angstrom. And that of the bound complex has also been determined to be around 17 angstrom. As you can see, as we approach 22 angstrom, we start to notice interesting changes among the energy profiles, where the complex CAM plus the white type has the lowest free energy as compared to the complex between CAM and between CAM and the triple mutant peptide. So this means that the complex calmodulin and the wild type is more stable than the complex with with mutation. So to gain more insight, we also added a new order parameters here, the probability of contact at the interface between the calmodulin and the benzene peptide. As expected, you can see that the complex calmodulin and the peptide with triple mutation is highly reduced as compared to the stability of the complex between the calmodulin and the wild type, or even the complex between the calmodulin and the system with IAK, which is one amino acid mutation. Then, so this is exactly the area where we have the encounter between the calmodulin and the peptide, and also where both undergo multiple conformations and the stable complex is formed. We then look at the contact map at this specific area to gain more insight. As you can see, new contacts are formed within the domain of CAM in the presence of the peptide with triple mutation, where those contacts were not seen in a complex calmodulin with IAK, which is one amino acid mutation, or even the complex, the CAM and wild type peptide. But still, this does not explain why we are observing such a large change in the calcium release rate from the C domain of calmodulin. We then cluster the structures from this specified area, and we visualize them. And here I'm showing the 10 most populated structures from the 25,000 structure for each complex, where we colored the representative structures by the root mean square fluctuations for each residue. I do want to say here that the T color here, residue with T color are the residue with less fluctuation, and residue with the magenta are the residue that show high fluctuation. As you can see, the residue in the C domain of CAM, as well as residues in the N terminus of the peptide, show high fluctuation. This could explain why we observe such a high high calcium release rate in the C domain of CAM in the presence of the peptide with triple mutation. We then plotted this root mean square fluctuations as a function of the residue number, as you can see here. As expected, we can see that residue, we can see that the complex with CAM and the triple mutant peptide shows high fluctuations in the C domain of CAM, which is happening mostly also in the calcium binding loop, of CAM. So then we can from this observation we can say that these RRK residues initially transiently interact with the residue in the calcium binding loop of the calmodulin before the native contacts are formed. So also if we look at the complex between the calmodulin and the wild type peptide, we can see that here that the fluctuations is almost the same in the N domain or in the C lobe of calmodulin. This further shows the stability of CAM as compared to that of the CAM in complex with the peptide with triple mutation. So to conclude this work, calcium binding to calmodulin, actually, there exists reciprocal effect between the calcium, the calmodulin, and CAM-KANES2, where the calcium binding to calmodulin affect the affinity of CAM for CAM-KANES2, and also when CAM-KANES2 interacts with the calmodulin, it affects the affinity of calmodulin for the calcium. So we were able to use our cost-square model to provide the residual explanation of the observed changes in the C domain of CAM as it binds to the peptide with triple mutation. A plausible explanation from our observation is that the RRK peptide, the RRK peptide or the RRK amino acids, which are the charged amino acids, actually interact with the calcium binding site of the calcium binding site of calmodulin before the native contact between the calmodulin and the peptide are formed. So this transient interaction helps to strengthen the calcium binding site and to keep the calcium in the binding loop, which were not observed in the case of the peptide with triple mutation, since those residues are not charged residues. So from this, I would like to thank my supervisor, Professor Marc. I would like to thank my current and past group members. And I would like to thank Professor Neil Watson and all the members from the Ruiz CTBP. And this work is funded by the NIH. We are grateful for the support. We also acknowledge the computing resources from the University of Houston and Ruiz CTBP. I'll be happy to take any questions, and thank you for your attention. Yes. Yes.
You went quickly over how you actually describe the interaction of calcium with the surrounding. So I think there's electrostatic. So are you essentially just taking the charge, the calcium, and using it to neutralize things that are in contact? But that only works if they're very close, right?
So you're talking about the experiment, or?
Well, in the simulation.
Yes.
I mean, these.
Okay, the simulation. Mm-hmm. Yes, absolutely.
So, tell us more about what actually happened.
Okay, so that's a very good question. So I do want to say here that in our simulations, we model the calcium effect. So we are not able to study the calcium release from our simulation. So we just model the calcium effect. So what's happening is that so I first want to show you our model. Let me go back here. So what's happening is that we use these interesting properties for almost all the calcium binding site in every calcium binding protein, where they adopt this pentagonal pyramidal geometry. Then we realize that in that geometry, we have the negatively charged residues, which are usually between I think 6 or 8. What we did is that we assigned the calcium charges to those residues. So we distributed the calcium charge to those residues just to neutralize the medium. So this is what we did to take into account that calcium effect. Now, if we go back to
if the structure fluctuated,
no,
that just doesn't happen.
No, it doesn't happen. Yes, it doesn't happen. That's what you see. That's what you see here. Let me go here. That's what you see here. You can see that actually because this CAM is a very multi-purpose protein, if I can say it this way. It has the flexible part. It has the globular, it has the globular part. So the globular part, where here you see the N domain and the C domain. At the same time, it has the calcium binding site. So it's really a multi-purpose protein, and it's really challenging to deal with such protein because you need to take all those effects into account. Yes. Yeah. Sorry, please, can you repeat the question? Oh, here? Here? Yes. Yes. That's yeah. So yeah. So we went into that. Into the calcium binding loop of calmodulin, trying to look at that pentagonal biopyramidal geometry. We found out that this loop tree only has 3 negatively charged residues, in that loop. Yes. Right. So that's an interesting question. That's exactly why when when it interacts with the peptide, that RRK from the peptide, which are the charged residues, we then enter with that calcium binding site to neutralize the medium and keep the calcium into that medium, which leads to high affinity. And then now, when you don't have those RRK residues, you can see that that property is lost, and then the calcium will just quickly dissociate from that medium. Interesting question.
Hello, good morning, everyone. First of all, thank you for letting me be here. I'm going to talk about my PhD work, which is mainly focused on quantifying the diversity in immune repertoires. So as we know, our immune system is a complex machinery that works pretty well since we are often facing different viruses, bacteria, and so on, and we are not all the time falling sick. And the main key of this success is the diversity of the protein receptors that are expressed on the cells, on the surface of the immune cells. So the main source of this diversity is the generation process of this protein itself. This process is called VDJ recombination, and it's an stochastic process that accounts for random rearrangement of genes, plus nucleotide insertions and deletions, and that has the potential to create up to 10 to the 61 different proteins. So I'm going to focus on one type of immune cells that are called B cells. So as a result of this VDJ recombination, we have a protein that is functional but has low affinity for antigens. It is estimated that in our bodies, we have a repertoire. When I say repertoire, I mean the set of cells with different receptors. So we have around 10 to the 12 different of these kind of receptors that are called IgM. Then, as I said, these receptors are not very antigen, has a low antigen affinity, but when they encounter this pathogen, they start a Darwinian process called hypermutations that is going to change the amino acid content of the protein to make it the binding stronger. And then we are going to have a new kind of immune cells that are IgG, or what we commonly spoke like antibodies. So these last years have been a revolution in immunology because we have developed a high throughput sequencing method that let us look very deep inside the repertoires of people. So from blood samples, we can get all this list of receptors in every person. So with these advances, we have we have seen these repertoires in big detail, and as I told you, with these big numbers, we have like 10 to the 12 different sequences. One may expect that when you look into repertoires of different people, these repertoires should be completely unique and private, but this is not true. So when you put together different people and different repertoires, we see that there is a non-negligible amount of receptors that are shared. So what I have done is to develop a statistical model to account in healthy individuals how many of these receptors will be shared. And to do so, we need to focus on the main sources of this convergence. So first of all, as I told you, this VDJ recombination, it's a main source of diversity, but the receptors that are generated, they are not all equally likely to be generated. Some of them, there is some statistical biases in this process that make some receptors to be very easily generated, and they will be present in almost all people. So what I use is a probabilistic model called PGen that from a sequence can learn the probability to be generated. And then from a sequence, once it is generated, from the pool of sequences that are generated, we are not sure, I mean, this sequence could be or not be selected. So we, on top of this first probabilistic model, we developed a second one that is called P-post, that from the features of the sequence, I mean, the length or the amino acid content, I can learn a selection factor, that is this Q, that ponderates this initial PGen probability, and it's gonna give me a final probability that is P-post. So the P-post of a receptor is the probability to be generated and selected. So now we are gonna try to infer this convergence among repertoires, and the main tool we are gonna use is the sharing distribution. When I say sharing distribution, I mean I took 10 different people which are healthy at the moment of sequencing, and I compute the list of these protein receptors, and for each sequence I count how many people contain the given sequence, and this is going to be the sharing number of the receptor. So from these 10 people, we have sequences, most of them are completely private, so they are owned only by one person, but then we have also sequences that are completely public, and they are present in all the individuals. And okay, so these crosses are the experimental data, and then we use the formalism of the generation function of probabilities to get a theoretical estimation of this distribution. So in this formula here, as you see, we only need to know the number of sequences that are in the repertoire, and then we need a probabilistic model. That is the model I told you about. We can use in this P, if we use the initial PGen model that only accounts for convergent recombination, we see that we underestimate the number of sequences that are shared. But if we include selection, we have a very good approach. So this analysis so far is done on IgM. There are these initial sequences. Then, as I told you, there are mutations that make things more complicated. And I worked on IgG also, but since at the beginning, I didn't include mutations, what I did is to take all these clonal families that are expanded, that are mutated, and I cluster them into families that are the ones that have the same genes, and also the 90% of amino acid similarity. And then I reconstruct the phylogenetic trees. From the reconstruction, I can remove the mutations looking at templates of genes, and then I recover the ancestor of this family. So the unmutated ancestor. And then I repeat the same pipeline, and it seems for IgG without mutations, it works pretty well. So far for healthy people, this model seems to work pretty well, but then we push the model a bit further, and we try to see if the statistics are skewed once people have undergone a common infection. So we took 44 people that were positive in COVID-19 at the moment of being sequenced, and we compute the sharing number distribution here, and then we use our theoretical estimation as a benchmark that should overlap, but as we see, when these people are having a common condition, there are many more amino acid sequences shared than what it is expected. So we are going to use this P-post distribution again to try to isolate the sequences that are overshared because they are responding to this COVID infection. So to do so, we compute, we compute two different probabilities. First, we compute this theoretical probability p-post I told you about, and then we compute empirical probability that comes from maximizing the posterior distribution. This probability is like the most likely probability that a sequence should have to explain the frequency on the data. So when p-post is a good probabilistic model, these two empirical and theoretical distribution should be the same. But for those sequences that are here in red, these outliers, p-post is no longer a good description because there is a selection acting on it, and acting on these sequences, and these sequences are probably like potential candidates to be SARS-CoV-2 response. So we took all these red outliers and we did some analysis. We compared them with an antibody database, and we found that there were more than 100 overlap with SARS-CoV-2 antibodies. And then we used them also to develop a log likelihood model to predict if the presence of these sequences in the repertoire of people can be determinants of this person having COVID or not. And it worked pretty well because, as you see, the log likelihood for people, I mean, there is it's very, the distribution are quite separated, so we can always know when a person is positive or negative in COVID because of the presence of these sequences. Well, as I told you, this is done without taking into account mutations, but then we need to know how mutations are acting and if it's increasing or not the diversity of the repertoire. So this is a work I'm doing now. This is in progress. My first approximation was using former work of a colleague of mine. This work it's a model that accounts for the probability of mutations happening depending on the context and the position in the sequence. So my main goal here is to be able to reproduce like synthetic data that follow the same mutation distribution than in real data. To do so, I take phylogenetic trees from clonal families of real data, and I reconstruct all of them. These phylogenetic trees have a branch length that is equal to the number of mutations that are happening. And then what I'm doing is to synthetically, with this p-post distribution, generate the ancestors and then induce mutations following this same structure that I can see in the phylogenetic trees. These mutations are induced using the model of my colleague that has 3 parameters. This gamma parameter takes into account the probability of mutation happening in because it depends first on the context. We need to, a mutation is more likely or not to happen depending on the nucleotide motif that is here. Then we have another parameter that it's accounting for the position, because there are hot spots and cold spots, and mutations don't happen like are not equally distributed along the sequence. And then I added a 3rd parameter that it depends on the length of the distribution. So this is the first ratio, and then we need another ratio that once we know where the mutation is happening, we need to know which kind of transition in the nucleotide level is taking place. So I induce the mutations following this distribution and the trees, and I got these synthetically generated sequences, and I compare with the sharing distribution, and for the moment it seems to work pretty well. But now I want to learn, this is done at the level of nucleotides. I would like to know what is going on at the level of amino acid. So it's my work right now. And well, this is all. I think this work is quite important because antibodies that we have been able to isolate using this model has been antibodies that has been elicited by the immune system of different people, meaning that they are probably easy to generate and they are potent, so they are very good candidates to elaborate some therapeutical vaccines or treatments. And also these antibodies can be used to develop some kind of test to know if a person is or not sick. And we could, I mean, what I would like also is to try to apply the method to other conditions and other infections and create some kind of database on potential antibodies responding on a different disease, and to know from blood samples which infection a person has undergone through his life. And well, that's all. I hope you enjoy, and thanks for the attention. Mm-hmm.
So I appreciated the agreement you got. It's very good.
Mm-hmm.
But that's a relatively simple curve. There's some independent prediction that your model makes, not the data that you develop about.
What do you mean with independent prediction?
I mean, how do I know that you're not just like fit the data?
No, but well, when I'm using this formula here, this is a prediction because I'm using what I'm using here is a people's distribution that doesn't. No, I mean, has nothing to do with the real data. The only thing, I mean, I'm using a curve of probabilities, and I'm summing over this curve of probabilities, but I'm not using any previous knowledge from. But the people's is universal. It's not learned on the data I'm working on. It's already a model. Yeah, I'm not. Yeah. Okay.
So I followed this for a while, but I guess I'm confused about this right now. When you do a calculation like this, do you know that the particular sequences that are shared are going to come out right? There's a sample.
Yes, exactly. Yes.
This
Yeah.
curve is some feature of shape of the underlying distribution.
Mm-hmm. Yeah.
But it might be that in any particular sample, the actual sequences that are shared,
Mm-hmm.
if you did the whole thing again,
Yeah.
the curve would look the same as the actual.
Yeah. I mean, this is also a problem that I'm trying to, like, I would like to look into it, because sometimes I feel like sharing, it's more about sampling. So if the sequence is very abundant, yeah, of course you are gonna find it. Or if you seep in more depth, you sequence in more depth, you for sure are gonna find more sequences shared. But yeah, right now, I mean, I don't have an answer for that, but it's for sure a problem I would like to go to. I mean, to see if there is some relation among sharing and abundance of the sequence in the repertoire or not. Yeah, no. I mean, this bias should be included in the probabilistic model itself. Yeah, because yeah, this PGen should account already for the bias on this sequence to be generated or not. Also, in this N that is taking the repertoire size, it should be included the at least the first approximation of the bias of sampling problem. Yeah. Mm-hmm. Yeah. Yeah, well, it's different. I mean, when we talk about T cells, the selection is when they are generated, they go to the thymus, and then there are like a negative selection that you remove the sequences that are reactive to your own proteins. For B cells, it's different because B cells, well, it's a different process, but there is some kind of selection that is called central tolerance. And it's more about antigen affinity than like reacting to your own body. Yeah. Well, I think, well, I'm not very sure because it's not me who developed these peoples, but in general, this selection can be seen in some features, that is the length, also some kind of amino acid composition that makes the sequence more likely to be selected or not. And I don't know if there is another parameter, but it's something that depends, yeah, most of it in the amino acid content of the sequence. Yeah.
Okay, thank you to the organizers for throwing together this event and for giving me the chance to speak today. So I'm a PhD student in physics at McGill University, which is in Montreal, Canada. So I'm a theorist, but I work in close collaboration with immunology labs at the National Institutes of Health in Washington, D.C. In particular, the lab of Grégoire Altan-Bonnet and his PhD student, Suraj Achar. And I also have worked closely on this project with Thomas Rademacher, also a physics student at McGill, who has now graduated. We also collaborate with labs of Naomi Taylor and Paul at the NIH. The goal in the project that I'm going to tell you about today was to establish a framework to quantify T cell activation in response to various antigens. So of course, this has been a problem studied previously, but we tried to shine a new light on it. So I'll start by introducing a bit this process of antigen recognition by T cells. Then I'll explain a few results that we obtained about this, in particular the cytokine dynamics of T cell once they are activated. And I will end by talking a bit about applying what we have learned on a separate problem to go back to mechanisms. So let's start with antigen recognition by T cells. So Maria told you about B cells. So T cells are similar. There are also white blood cells in your blood and lymphatic system that are constantly scanning the surface of other cells, looking for traces of pathogens. The T cells have something called a T cell receptor, which is in some ways similar to a B cell receptor, but in some ways also different. So it's not an antibody, it's really a receptor that stays on the surface of the T cell. And just like B cells, T cells, they mature. In the case of T cells, they mature in the thymus. And they are selected so that they never respond, when you grow up, they are selected so they never respond to self-antigens or antigens from your own body. But they have receptors that are then specific to foreign antigens, so fragments of pathogens. When a T cell scans the surface of another cell, if it encounters the antigen to which it is cognate, this will trigger a series of reactions in the T cell, and there will be it will detect it by a slight difference in binding time between the T cell receptor and the antigen, a factor of 2 or 3 times the affinity, and that's enough to really trigger proliferation of the T cell, production of cytokines, the activation of effector functions to kill infected cells, and so on. So as you may have understood, T cells, they should not respond to even large amounts of weak antigens, so here on the top left, but they should be able to respond very specifically and fast to even small amounts of strong antigens. And the difference in binding between weak and strong antigens is not that large. So there has to be some non-equilibrium amplification process. And in fact, there's a very complicated molecular machinery in the T cell to ensure this sensitivity and specificity. Here I'm showing the signalosome of the T cell receptor, which is in the middle, that's the peptide. And there's a bunch of other molecules recruited at the CCR upon engagement. So that looks very complicated, very intimidating to a physicist like me. But fortunately, it's possible to find simpler models that explain the basic effect we want here, which is that T cells respond strongly to even small amounts of strong antigen, like the curve in red here. The x-axis is a number of antigens. and they never respond even to large amount of self-antigens. And it has been proposed that kinetic proofreading does the job, and shown also that if you use something called adaptive kinetic proofreading, where you add one negative feedback to the proofreading chain, you can really ensure that there's a good discrimination between self and non-self on a large range of antigen abundances. So the negative feedback makes the response of that network mostly independent on the amount of antigen and make sure you only respond to foreign pathogens. And so that's an example of what this model gives. You always respond to the strong agonist, but never to the self-agonist. But now if we go back to my first slide where I've showed that T cells produce a vast array of cytokines. Here I'm showing the 5 that we have considered in this project, but in fact they produce over 30 in the body. Why would they have such complicated extracellular messaging proteins? So these cytokines serve to alert other immune cells and send messages to other immune cells. So why would they need such a complicated code if it's just to say they're self or non-self? So we asked the question, is it really just a binary decision that T cells make? And are these cytokines a code for the readout of antigen quality made by the T cells? And this is the topic we tried to address in this work that was published 6 months ago and done in close collaboration again with Suraj in Grégoire's lab and with Thomas at McGill and supervised by Paul and Grégoire, of course. And we were interested in particular in antigen quality, not just for its own sake, but also because it's relevant to current research in cancer immunotherapy. I'm going to give 2 examples here. The first one is that clinicians are trying to develop cancer vaccines where they sample antigens from the patient's tumors, inject them back later on in the patient to create an immune response from the patient's own immune cells against the tumor. And an important step in that process is to select the best neoantigens, so the best antigens from the tumor, based on their quality, but the methods used are often cheap, but not super cheap in the sense easy to do in a lab, but not very accurate. So we think there's room for improvement there. And another example is that antigen quality is a good predictor of how successful immune therapy will be in patients. Here I'm showing, for instance, results of a model where patients are classified according to the quality of their neoantigens, and those with a higher quality antigen tend to survive better after treatment. So that's why we really try to understand how antigen quality controls the production of cytokines by T cells. The first part of our project was uncovering a latent space of how these cytokine dynamics are organized. So I'm going to explain that in a moment, but first we needed data, and we were very lucky to collaborate with Grégoire. In their lab, they have developed a robotic platform where they can track the activation of T cells over time in a vast array of conditions. What they do is they prepare co-cultures where they mix T cells on the right with antigen-presenting cells loaded with various antigens present in various concentrations. So they can test all these conditions and then measure the concentration of many cytokines over time in each of those co-cultures. They also have developed a processing pipeline to analyze the large amount of time series that come out of this platform. And then they can share that data with us. And what you get in the end, let's say you run a few experiments with many co-cultures in each experiment, you get an array of a large matrix of cytokine time series where the time series are caused by different antigens, here denoted by the different colors. And that can also be present in different concentrations, here denoted by the different line styles. So we have this matrix of data. It's somewhat noisy. There's overlap between cytokine time series coming from different antigens. You can see some segregation according to color, but except for the blue one, it's hard to tell what's happening. And we wanted to know exactly how are these dynamics controlled by antigen quality. The first thing we did as theorists was to pre-process a bit further the data to remove noise, to smooth it, to interpolate it. But we found that first taking the logarithm of cytokine concentrations was a useful trick to both reduce the noise and see better what's happening, because these cytokine concentrations vary over multiple orders of magnitude, so log scale is better for this. And we also found that looking at this quantity here, the cumulative time integral of the log of cytokines, allowed us to separate a lot better the trajectories according to antigen quality. Of course, that was our goal, and I found this trick during my master's thesis, and part of it was looking at how we can process that data to make sense of it, and using different unsupervised methods and supervised methods. I found that this time integration trick was already helpful. So we took that for granted for the rest of the project. And then Thomas asks, can we reverse engineer those time series? Can we in particular, if we're given a certain amount of cytokine, predict which antigen caused the production of that cytokine in the platform? And he trained a very simple neural network to do this job, where the input to the neural network is the concentration of these cytokines measured by our collaboration, and more particularly the time integral of the log of cytokines. There's one intermediate layer in the network, which has 2 dimensions, and the output layer tries to predict which antigen was the source for the cytokine received as input. So we trained, Thomas trained the network. He got good classification accuracy on test and training sets and so on. But really the most striking feature to us was not so much the fact we could do this classification task, but rather it was the structure of the data, what it looks like when you project cytokine time series on these 2 nodes here. And that's what I will be calling the latent space. It's a term from machine learning. It means a lower dimensional space in which you compress the data. And when you take many cytokine time series and project them into the latent space, you see this very striking structure where cytokine time series coming from different antigens, denoted by the different colors again, end up in very different regions of the latent space. And the curves have a pretty simple shape, as I will show later. But this was really striking that there's apparently a lot of information about antigen quality in that latent space. So we call this property antigen encoding, meaning antigens are encoded into the cytokine concentrations. And we see it better when we look at a 2-dimensional space. What's nice also with the latent space is we realized you can actually reconstruct all 5 cytokines considered here, just based on the position in a 2-dimensional space. There's almost no loss when you go from 5D to 2D in this case. Turns out we were a bit lucky because these cytokines have correlations between them, but we didn't know that to begin with. So the small machine learning algorithm told us it's actually a low-dimensional system here. You don't need all cytokines, you just need 2 dimensions. So once we had that latent space, it was great for us theorists because it's a lot easier to make a model in a 2D space than in a 5-dimensional space. It's hard to relate TCR engagement to all these cytokines, but it's a lot easier to just describe the simple curves I've shown you in latent space. So the first thing we did is we sat down and tried to find mathematical expressions that could describe the shape of those latent space trajectories, just by guesswork, the phenomenological, not based on mechanisms. We had different versions. The simplest one was inspired by the motion of a projectile in physics, where you have an initial velocity corresponding to how much cytokines you produce initially, time t0 at which consumption of some cytokines start, and an initial theta that corresponds to the ratio of the different cytokines at early times. What we did is we fitted those mathematical equations to each trajectory for the different peptides, different concentrations, to see how these parameters, V0, T0, theta, dependent on the quality and the quantity of the antigens. And again, we saw a striking structure where first, the various parameters, here I'm showing 3 of them, V0, T0, theta, are organized according to the quality of the antigen that caused the time series. And there's only minor effect of the quantity of antigens. And moreover, all parameters are correlated. So it means we're not very efficient in our model because in fact, it's a 1-parameter model. You need 1 parameter to describe those curves. And this parameter is, as you can see, antigen quality. What's nice also, once you have mathematical equations for the time series, the time dynamics, is you can generate the new time series by just sampling parameter values in the space of parameters corresponding to the different antigens based on fitting on the previous, based on looking at the distribution on the previous data set. Then you can compute relevant quantities in the latent space and use these latent space quantities to reconstruct all 5 cytokines. The reconstruction algorithm was something, I think it was quadratic regression, where we fit the coefficients on a separate data set and then fix those coefficients and use them to reconstruct cytokines from model-generated latent space trajectories. So it's nice you can generate cytokine time series, and we think it will be useful for us, at least in the future, to continue studying, let's say, how cytokine receptors work now. We have synthetic data, so we can do different things with it. The last question we asked was, how much information is there really in that latent space? I've shown you there's Antigen encoding, the trajectory is separate according to quality, but can we put a number on how good the encoding is? So we thought of our latent space as an information channel where there is information between the quality of the antigen, Q, and the model parameters, theta, fitted on the latent space trajectories corresponding to those antigen qualities. We realized that the amount we could get from that calculation was limited first by the distribution of peptides available experimentally. We only have a finite set of peptides we can test. And this means we don't necessarily fill optimally the parameter space here. So we designed a procedure where we fit the parameter distributions for experimental peptides and then interpolate these parameter distributions as a function of peptide quality, such that we can now generate parameter values for arbitrary antigen qualities, even those that are not present in the training set of peptides. And this allows us then to optimally fill the parameter space, such as we can maximize the mutual information. And in fact, we computed channel capacity, which is mutual information maximized over the arbitrary distribution of the input peptides. And when we did that calculation, we found this number, 2.6 bits, which may not seem like super interesting, but actually it's super relevant for immunologists because they think of peptides as self versus non-self, as I have introduced in my own talk. Sometimes they think about three categories related to thymic development, but 2.6 bits tell you that you can subdivide the continuum of antigen qualities into at least into something like 6 classes. And if we had tried even more perturbations, maybe there's even more information we can put in that latent space. What this means is that T cells have a finer discrimination power between antigens than just a binary decision. And we think this will be important for immunologists. And now we had that latent space, simpler description of cytokine dynamics. A lot of it was based on fitting data, looking at the fitted distributions, making a few predictions, generating synthetic data. But can we go back now to mechanisms, to what's actually happening in the T cells? We had an interpretation of our latent space model, but can we be more explicit in terms of biochemistry? So that's what we try to do here. We have an interpretation for our latent space variables. First of all, here I'm showing what they actually are. Latent space 1 is basically made up of the cytokine IL-2, and latent space 2, the other axis in our latent space, is a difference between these two cytokines here, which I will describe as adaptive cytokines, because you're not all immunologists, so I can make that approximation. And the other three cytokines, which are subtracted, are more like innate cytokines produced by all kinds of immune cells. IL-2, IL-17 are more specific to T cells. So that's a difference between adaptive and innate cytokines. And when we look at how these latent space variables behave as a function of antigen quality at a certain time point, so you take all these trajectories at a given time point, you see that LS1 only increases, so that's like the activation of the T cells, that's IL-2 basically. But LS2 first decreases and then increases back up. And this is a sign that actually these innate cytokines that I've talked about are activated more easily by the weaker antigens than the IL-2 cytokines. So innate cytokines activate faster. And we think, so that's really a hypothesis, but we wanted to see how far we can do with that hypothesis. We think this comes from an effect of the negative feedback in the model that I've talked to you about. So that feedback would affect IL-2 more strongly for weak antigens, and the innate cytokines would still be active. So that would give you a net negative LS2. And so basically that part of the LS2 variable where it's negative would be a sign of negative feedback on adaptive cytokines. We made that hypothesis, and we tried to see what it tells us then when we look at T cells that have an altered T cell receptor. So collaborators in the Paul Love's lab at the NICHD have engineered a mouse line which has fewer phosphorylation sites on its TCR, so that might change its function, and actually it does. And we try to predict in particular, based on our hypothesis, how does this effect called antagonism change in the altered T cells. And antagonism is the fact that if you take a mixture of antigens, the response to that mixture can be lower than the response to a single antigen. So if, for instance, the T cell responds, as shown in green here, the amount of IL-2 it produces, it responds like this to a single antigen called N4. And then you try to activate that T cell with the same amount of N4, but on top of that, a large amount of this weaker antigen V4. The response of the T cell can be actually lower. Even though there's more peptide overall, the addition of the weak peptide can reduce the response of the T cell. So it's not additive at all. And that's what we call antagonism. So we took these T cells with altered receptors. Our collaborator, Suraj, did that. He ran the experiment on the platform. He tracked their cytokine production over time in response to various antigens. And we projected that data into the latent space as before, and we saw interesting patterns. LS1 was shifted to the left, meaning that these T cells are more activated by weak antigens. And moreover, LS2 lost this negative region that I told you about. It also became monotonically increasing. And this based on our hypothesis, this told us that these altered TCRs have a weaker negative feedback. There's less negative feedback on them. So we implemented those two insights in our simple kinetic proofreading model with a few bells and whistles that I can tell you more about later if you want. But basically, we reduced the amount of negative feedback. We reduced the number of proofreading steps, so it's more responsive to weak antigens. And we made predictions then about antagonism based on that model. So in particular, we looked at the ratio of the response to the mixture divided by the response to the agonist alone. And if that's lower than 1, it means you lower the response, it means there's antagonism. And qualitatively, at least, the patterns predicted by the model matched the data. First, we indeed saw less antagonism with the altered TCRs, and we think that comes from a reduced negative feedback. And we also saw other effects that were confirmed by experiments, like the fact that the best antigen to antagonize the response to a strong agonist changes as a function of the concentration of the agonist. So you can see a crossover between those two lines coming from different antagonist ligands. So we were quite happy with this, but we're following up now on these results and trying to make even more mechanistic models of antagonism like this, in particular in CAR T cells. So that's work to come. Just to summarize, we're very lucky to have access to robust, abundant cytokine time series from Grégoire's lab and their robotic platform. This allowed us to find a latent space structure underlying cytokine dynamics. And in that latent space, we saw a property we called antigen encoding, where antigen quality, so the strength of the antigen, could be subdivided into 6 non-overlapping classes, meaning there's a lot more than binary decision that's being made by a population of T cells. And we used insights gleaned from that latent space to make mechanistic predictions about antagonism in T cells that have altered receptors. So with that, I want to thank my collaborator, Grégoire, my supervisor, Paul at McGill, and my fellow co-authors, Thomas at McGill and Suraj, who did all the experiments and developed the processing pipeline. And also everyone in Paul's lab, in Grégoire's lab, and also Naomi Taylor's lab at NIH and Paul Love's lab at NIH as well. So thank you for your attention. I'm happy to take questions. Yes?
The T cells only talking to each other with the cytokines? I mean, is there, how much is cell around?
So the question was, are the T cells also talking to each other through cytokines? And definitely there's, first T cells produce cytokines for themselves, but they also produce it to co-opt other T cells into responding. With the platform we have, we were not able to completely disentangle the two effects, but definitely T cells have receptors for cytokines. So the T cells that would be a bit less activated maybe by the peptide would be then prompted to activate more. There's also dynamics where T cells first produce cytokines and then consume them, and that gives the increasing then decreasing shape of some of these cytokines in particular. So there's definitely influence there. We're trying to disentangle that with mechanistic models now, but from that project, it's hard to tell exactly what's within the T cells and what's a population effect. Yes.
When you do the calculation channel,
yeah.
In some way, you have this prediction of something about the distribution, and this is there, can you imagine testing that? You you chose only some inputs.
Mhm.
But then you get a prediction for what. Is there a way of sampling at random from the you understand what I'm getting at?
Yeah, trying to see if there's optimization of this channel such that it's tuned to the natural distribution of these peptides. Yeah, it's hard for me to tell because again, we're mostly testing the distribution here in our experiments is fixed by how many times we try each peptide in the lab. So that's hard. And then we see the optimal distribution. Of course, there's edge effects where you put a lot of weights to the extreme cases, self versus non-self, but we see also intermediate peaks. I think the natural distribution is pretty different from that, actually. Most antigens recognized by T cells will be self, so it would be more skewed to the left. So I don't think this says something about the optimality of the channel, unfortunately. But we thought about this. I don't have a solid answer. But I wouldn't go as far as interpreting this as some kind of optimality principle. Yeah, it's hard for me to tell. It's disappointing, I'm sorry, but I don't really know.
Can you use this as an experimental prediction? So maybe the answer is you can't make it.
It would be hard to go in the body and see the frequent, I'm not sure. We could discuss that more if you have ideas, but I don't really know what to make out of it. Yeah. Yeah, definitely. Yeah, definitely. And yeah, and I actually think a single T cell is closer to having binary responses. I think we did single cell quantification. That's what they're doing now in Grégoire's lab. I'd say it's more than binary, but you definitely have typically two peaks that appear. But you have a lot of T cells also in the middle. So it's a population response, yes. So this is in that project we mostly looked at a specific set of peptides which are well characterized. They come from I think they come from bacteria, actually chicken ovalbumin. So there are a set of single point mutations on one peptide that is a strong agonist for a certain T cell receptor. So I would say they're bacterial or viral. I don't remember, but we didn't look at neoantigens in that project. We're doing that in other projects, but not in this one. So it's just a set of well-characterized, if you want, model peptides for immunologists. Yeah, it's different depending on the pathogens. It's also different depending on which cells are presenting the antigen to the T cells. So here, all the data I've shown, I think, was done with the antigen presenting cells are splenocytes. So basically anything you can find in the spleen. I think that would be more what you see when you have a viral or bacterial infection. For worms, I'm not sure what you would use, but splenocytes, the antigen presenting cells. So I should say though that Grégoire and Sourage looked at other antigen presenting cell types, and we saw a similar structure in latent space. So it seems to be quite conserved, actually, the way these adaptive and innate cytokines combined to form this latent space. Thank you.
Yeah. Thank you a lot for having me today. I'm very glad to be here. And I'm very excited to talk to you today about our efforts in developing a theory that, for cell size homeostasis. And if you, as you can see in the video that's playing, that cells as they grow and divide in this process. So this video is of École. So in this process, cells are subject to a lot of noise sources. Some are internal due to processes inside the cell. Some are external due to environmental factors. So the cells must have a regulatory mechanism to stay a stable system and to control something very important for its functionality, such as size. And experiments such as the one I'm showing a video to, it's very hard to track the progression of cell size across generations. So a decade ago, a very innovative microfluidic device was created called the mother machine. And in the mother machine, you have a mother cell that's trapped at the tip of this road-shaped trap. And when the mother divide, you can measure the size across generations as shown in the picture to the right. So with each division, you measure the birth size of the mother. So you end up with data like this of a single lineage measurement of size across generations. And you can see that the data are, the size is distributed around some mean. So this is the data that I'm going to be working with today, trying to develop our theory. So more specifically, the structure of my talk will be, first of all, I will talk to you about a standard model that exists currently in the literature, and then our efforts addressing some problems with the model and trying to improve it. So specifically, the two things that I'm going to talk about is the first, the prediction of short-lived correlations, which is a measure of memory in this system. So the first part, it will be phenomenological. Then the second part, I will get more mechanistic and try to propose a molecular mechanism and see how it affects the dynamics of the system. So to start with the model, that's currently used widely in the literature is based in two observations. The first one is just the observation about the exponential growth of bacteria. So as you can see here, cells started from a birth size Pn will grow exponentially with a growth rate alpha n, and then divide as the time Tn, with a division factor Fn here, which is on average 1/2. The division factor itself has its own noise. So this way we have an equation that relates birth size across generations. The second observation is an observation about correlations between the growth factor alpha Ntn and log of the ratio Bn over the ratio between the cell size and the average cell size. So, and you would expect that this correlation be negative, because if you look at this equation, that means if you grew a little bit above your target size, you would want to compensate this with just having just less or more growth factor. So I'm going to rewrite these two plots here in another form by making these definitions. So the first one is the log of this ratio, I'll call epsilon n. The second one is delta n, with just the growth factor minus its average. And these both factors are both measures of the deviations from the average behavior. So epsilon n will be a measure of the deviation of birth size from the average, because if you're exactly at the birth size, epsilon n will just be 0. You didn't deviate. If it's positive, that means you're above the average. If it's negative, it means you're below. And the last thing, I will just introduce a noise factor associated with the division process. So these two equations here summarize these two observations and what I just said. The only thing is that I will just add here a noise that affects for the noise that's associated with this growth process. So equations 1 and 2 now is what I'm going to be calling the standard model of size control. And it turns out that this correlation factor beta has a deeper meaning for the dynamics of the cell. The first one is beta equals 0. It turns out that beta equals 0 is what's called a timer mechanism, meaning that the cell wants to divide after after a specific time. So as when the cell is born, it will grow for a specific time and then divide immediately. The second one, beta equal 1/2, is what's called an adder, that the cell, regardless of its birth size, will just add a constant size and then divide. The last one is beta equal 1, which is a mechanism that's called a sizer, that the cell will just wants to grow to a target size, and then it will divide when it reaches exactly that size. And we can make predictions about things like the correlations here. So we can calculate the correlation from the model, and we find for an experiment like this where they calculated beta equal 0.6, for example, and it's in very good agreement with what we see in the data. However, a few years ago, another microfluidic device was invented called the Sisters machine. And in the Sisters machine, instead of having a mother cell in a rod-shaped trap, you now have a mother cell that's trapped at the tip of the Sisters machine. Then it divides into two sisters, with now each side of the trap having its own dynamics. And the reason they did that is that they wanted to remove away from the system the effect of external noise in the system, because the effect of the two sisters would be similar to, so when you compare the two sisters, which are genetically identical, you can extract information about the internal behavior of the cells, excluding factors from the environment. And if you look at the middle, you'll see that the size of the two sisters, how it progresses with time, and they stay correlated for a long time, and they calculated the cross correlation from this. So now this gives us a new prediction for the memory of cell size. And when we look back in the literature and try to calculate this prediction from the standard model, we find that the theory informs us that the autocorrelation decay time scale should be longer lived than the decay time scale of the cross correlation. However, what we see in the experiment is the opposite, that the memory is actually longer lived than we previously knew from the mother machine experiments. So we wanted to account for this behavior in the theory. So we took a step back and we looked at the individual autocorrelation data, where you get from the individual traps of the mother machine, and what you see is that different dynamical behaviors of different lineages. Some are oscillating, some are just decaying, and when you average them, like you do always in any any mother machine experiment to find the autocorrelation, you will find this average decay behavior. So we hypothesized that these different behaviors of different lineages are actually indicator that something is causing these lineages to have different dynamics. And even though microfluidic devices, the environment is always assumed to be constant, but we believe that the interactions in the microenvironment of the cells with their environment can create differences in the dynamical behaviors of cells. So we tried to account for this behavior, and we did two things. First one is our model parameters should be different for different lineages. And we added a new parameter lambda here, which tries to account for the memory we observe, which is just the feedback from the growth factor on itself. So this model here is a discrete version of a damped harmonic oscillator that's enough to capture different dynamics we observe. Some are oscillating, like you see here, some are exponentially decaying, and these two regimes are the most important ones at what we see in the data. And after that, we made a prediction about the autocorrelation, calculated the autocorrelation here, and fitted the autocorrelation to the data from the system machine. So in gray here are the theoretical autocorrelation calculated for different lineages in the system machine. And we took the parameters that we fitted from here and we calculated the PCF was, and our prediction was we should find this time scale separation between the autocorrelation and the cross correlation when we calculate this. And as you can see, when we compare with the experiment, we find that this introducing this variability between parameters and accounting for these oscillations is enough to create this separation between timescales and autocorrelation. So now this was the first part of the talk where it was all phenomenological. Now I want to highlight the second problem and introduce a mechanistic model. Now, the second problem with the model is that it neglects noise associated with internal processes that's happening inside the cell. So for that, we added to the model. Before going to the model, I just want to go back to the standard model and the prediction it makes about something like size control. So it makes a prediction that size noise would just be maximum here at the timer behavior and then decay to a minimum at the sizer behavior for beta equal 1. So we wanted to introduce a molecular mechanism for this. So we just proposed a very simple mechanism that consists of a protein X with this protein X growing to a threshold X star. And then when it reaches a threshold X star, it will immediately divide. So this is like saying that the cell doesn't control size directly, it controls something internal instead. And then when this is satisfied, it divides, as you can see in the figure to the right. And this X actually have several candidates that were proposed in the literature. First one is a protein called FTSZ, which is important for the formation of the division septum. Something like DNAA is also a candidate. Peptidoglycan is also a candidate protein. So there are different sources that are saw this behavior before of a protein growing to a threshold. Now, in different limits of this model, we are able to get back the timer, adder, sizer mechanisms that we see bending on model parameters. And this is a heat map that explains this, that when you calculate the beta parameter, which is depending on its value, it can be an adder, sizer, or a timer. In different limits of our model, we're able to recover back the mechanisms. So we want to see how this compares to what we had before and how the prediction would change. And what we find is that in the standard model, it just neglects the dependency on any internal mechanism. So you get this just one line curve, but instead, when you introduce a mechanism and you take into account that your division and size control mechanism will depend on model parameters, which is beta here. As you can see, it's now a function of the dimensionless parameters of the model. And also that the noise factor associated with the growth factor is now also dependent on the model parameters. And most importantly, you see that the minima actually shifted from 1 to 0.5. So there's a new prediction in which mechanism will have a minimum size. So we simulated this model to see what's going to happen. And we see that this envelope theoretical bound curve is satisfied. And you can imagine each point of the simulation here as being a different experiment with a different set of parameters and conditions. So we obtained data from 6 different sources and to see if our prediction for this model be satisfied. And what we see that the data follows this prediction of a bound. But also what I want you to take from this slide is that This points out that that there's a change in the minimum. It's no longer at the size or beta equal 1, but it's now at the middle. But also, as you can see, the data are smeared, and this points out that it might not be the case that cells care about size so much, because if it was the case, you will find that all data lying around this bound. But instead, this points out that cells are probably controlling a different mechanism inside the cell. That's why we see this smear behavior across mechanisms. So this takes me to the end of my talks. I wanted to summarize what I discussed today. The first points that we hypothesize is the different microenvironments correspond to different dynamical behaviors, and allowing for this oscillation behavior might explain the timescale separation between the cross correlation and the autocorrelation, and can explain the extended memory that we see from the sisters machine experiments. Also, taking molecular noise into account can change our understanding of the cell size dynamics. And I want to thank my advisor, Andrew Mugler, and our collaborators, Hannah Salman and Harsh. And with that, thank you for listening, and I would be happy to take any question. Okay, so your question is there a homeostasis mechanism for the growth factor itself, and not just size? That something else is controlled other than size?
Well, I think you said it.
Yeah, and I was referring to that there's a protein inside the cell, which I'm assuming to be X here, so something more mechanistic. And part of controlling size is also part of this is controlling the growth factor. And if I remember correctly, sources like the one I'm citing here also looked at correlation in cell cycle time, and they see very similar behavior to the correlation in size. Yeah, yeah, I see. Maybe there's another variable other than size, but I don't think it would be the growth factor, because it's also still not mechanistic to do something like that, or maybe a mechanism that can control growth division itself directly, not size, maybe, or I mean the division time itself, maybe. Yes. Yes. Okay. I didn't quite hear the question. I'm sorry. So. Yes. Yes.
You just go to volume just below the target volume, and then you will kind of, instead of going to the front, you kind of oscillate around this point.
I see.
And that looks a bit like the oscillation.
Introducing, so if you start at a size that's greater than the average,
yeah,
then if you have just below the average, yes. Yes. Yeah, so that's all the different mechanisms of sizer, adder, and timer. They are all they all cover behaviors like this. It doesn't matter whether you're above the average or below the average. If you are above the average, what will happen in the case of the sizer? Because once you're already below the target size, you're already above the target size. I'm actually not sure. I'm not sure how the cell would do, but maybe that's also a reason why in several data, mostly you don't have either sizer exactly, so it's never 1, and it's never also 0. It's always around 1/2. So maybe because some behavior like this will get funny. Yeah. The oscillatory and alternate dynamics was already in the traditional model. Did this appear experimentally, or is it a bug of the model? So the oscillatory dynamics was not part of the traditional model. No. The alternate dynamics, the model will be stable in the alternate dynamics. So this would correspond to beta going to greater than 1. So between 1 and 2, this will be also a stable part of behavior of the model will still be stable, but we don't observe it in the data. There isn't really alternation. So did this answer the question? Okay.
Are we good now? Okay, cool. Yeah, so I'm Matt. I'm from Simon Fraser University. And today I'm going to tell you a little story about the physics of molecular machines. So molecular machines are nanoscale protein structures, which are integral components of a whole host of different functions within the cells of every living organism on Earth. So they transduce between different forms of energy, be it electrical, chemical, or mechanical. They transport materials within cells. They assemble complex structures like proteins and nucleic acids. And they can perform logical operations, which allow organisms to interact with their environments. And now I like to kind of think about some defining features of these molecular machinery, or you can call them stylized facts. And the first, for those of you not familiar with the Reynolds number for fluid dynamics, it basically quantifies the ratio of inertial effects to friction effects for an object immersed in a fluid. And so plugging in some kind of reasonable values for a molecular machine like kinesin, which is about 10 nanometers tall, and moves at a speed of around 1 micrometer per second, we get a Reynolds number on the order of 10 to the -8 or 10 to the -9. And so what that means is that friction forces dominate over inertia. So all motion is overdamped. Inertia disappears pretty much instantly for these systems. Another interesting feature is that their dynamics are characterized by inherent stochasticity. So the energy scales of different forces driving the dynamics of these molecular machines are comparable to the ambient energy and thermal fluctuations, or KBT. What that means is that motion is characterized by significant stochastic fluctuations. And results that we have for macroscopic systems, like the second law of thermodynamics, where entropy production of the universe always increases, are only true on average for these microscopic systems. Now, my favorite feature of molecular machines is that they often work collectively to accomplish their tasks within cells. So to give a couple examples, the molecular machine ATP synthase, which turns a non-equilibrium proton gradient across the mitochondrial membrane and uses that to power the synthesis of ATP, which is the energy currency of the cell, is composed of 2 components, one of which interacts with an electrochemical concentration, the other does the actual synthesis of ATP. The molecular machine troponin, which regulates the turning on or off of myosin motors in cardiac muscle tissue, is composed of 3 interacting protein subunits. Photosynthesis in plant and bacterial cells is accomplished by many different interacting molecular machines. And an example I'm going to tell you a lot more about today is motor-driven transport within cells, where anywhere from 1 to several hundred transport motors can work together to transport a single cargo within a cell. And so these molecular machines really are machines in the way we use the word machine colloquially, but we can't think of them in the same way that we think of macroscopic machines. So large-scale machines like motors and steam engines and heat engines are well described by the laws of classical thermodynamics that have been around for centuries. But for these molecular machines, which behave stochastically and are far from equilibrium, we need a new theory called stochastic thermodynamics, which has been developed over the last 20 to 30 years and allows us to quantify the energetics of these kinds of systems, among other things. So in particular, we can think about nanoscale stochastic systems that exchange energy with work and heat at reservoirs to accomplish their tasks. But as I mentioned, most molecular machines are actually multi-component systems. So we can expand this kind of framework to think about systems with many interacting degrees of freedom. And because we're physicists, let's not get carried away. I'll expand this picture to now feature two interacting components within the system. So stochastic thermodynamics tells us how to assign interactions between these components of molecular machines with their environment, so they can exchange work with external reservoirs and heat with external heat reservoirs. But it doesn't tell us a whole lot, at least at the moment, about what's going on inside them. So we know these kind of coupled components of a molecular machine need to interact, but we don't necessarily know what's going on. So thinking of ourselves as like 16th century sailors making a map where we don't know what's there, we can just say, okay, here be dragons. So some of what I want to tell you about today is how do we think about things where we don't know how the components of a system are interacting? So there's kind of two approaches, one of which is to place fundamental bounds on the performance of these systems that are independent of that kind of here be dragons region. And the other is to try to use some tools from stochastic and thermodynamics to actually infer what's going on there. So the system I'm going to focus on for most of this talk is collective motor-driven transport, where motor proteins like kinesin or myosin or dynin transport molecular cargo around within cells. They synthesize or hydrolyze ATP to produce power-directed motion and walk along microtubules, towing cargo behind them. And as I mentioned, these motors work collectively with widely varying numbers of motors attached to a single cargo at a given time. So just to give you some examples, for kinesin pulling small vesicles, you might have 1 to 10 motor proteins per cargo. For myosin motors sliding actin filaments within muscle tissue, you might have on the order of 100 motor proteins. And for kinesin pulling large organelles like mitochondria within cells, experiments have found well over 200 motor proteins in some cases attached to a single cargo. So there's a really kind of wide range of possible numbers of components to these systems. And kind of taking a step back and just thinking about, suppose we have a transport system within a cell. What are some things we might like to optimize? So in particular, we can think, you want the ideal transport system to be fast. You want it to be precise so that it transports the same amount of cargo in the same amount of time every time. And you want it to be efficient, so minimizing power consumption. And to me, thinking about these collective systems, there's kind of two questions that naturally arise. So the first one, which we kind of took some steps towards answering in a paper earlier this year with me and my supervisor, David Svok, is how does the number of motors in these kinds of systems impact these different metrics of performance? And that's not what I'm going to talk about today. Today, I want to tell you about some things we can say from stochastic thermodynamics on fundamental limits that constrain their performance irrespective of the interactions between the different components of these systems. So to kind of make progress on this, we need a general framework for a model. So I'll tell you a little bit about the class of models we're thinking about. So we consider a diffusive cargo with some diffusivity coupled to an arbitrary number of motors. Each motor can be described by some diffusion coefficient and some kind of chemical driving force. These need not be equal or even in the same direction. And then the components of this system, the motors and cargo, are linked together by some kind of conservative potential. And we make basically no assumptions about what this potential is. So it might include terms for interactions between the motors and cargo. It might include terms for interactions between pairs of motors, or it might include terms for interactions between the motor and the substrate being the thing the motor walks along. In the case of kinesin, microtubules. So you might generally, for a free motor not attached to a cargo, see an energy landscape like this that's periodic with some kind of metastable states, energy barriers. We're just assuming that this potential exists, none of the details. And we can model this system using multipartite overdamped Langevin dynamics, which accounts for the stochasticity and low Reynolds number that are defining features of these systems. Just to give you a little bit, sorry, go for it.
This seems innocuous, but we're assuming that these things don't have any internal states.
Well, so yeah, we can kind of think about the, like as I've kind of drawn this periodic energy landscape that has different metal stable states, we can think of those as internal states. The assumption here is that it only has kind of one cycle that it moves through as it moves forward along the mechanical degree of freedom. Many of these results allow for kind of orthogonal degrees of freedom, but in a kind of first order coarse graining approach here, we're thinking like 1 degree of freedom where the mechanics and chemical degrees of freedom are tightly coupled. And for kinesin, at least, the tight coupling between mechanical and chemical degrees of freedom is generally pretty well supported. Although there are certainly cases where that assumption needs to be relaxed, and that's something we're actively working on. Okay, so just to give you some firm grounds to stand on, we can consider an example system where we just have 2 identical motors with identical chemical driving forces, some kind of sinusoidal energy landscape with a maximum force, which is the maximum conservative force due to this energy landscape. You can kind of think about it as the height of the energy barriers, quadratic coupling to the cargo, and no interactions between the motor. And simulating the kind of dynamics for this system, you get trajectories that look something like this, where here the light curves are the motor trajectories, the dark curves are the cargo, and the different colors represent different kind of heights of energy barriers in the periodic potential. And so as we go from like no energy barriers to large energy barriers, you see the motor dynamics kind of go from being sort of continuous to more like they're taking discrete steps, which is generally more like what's seen experimentally. And just to say, we can kind of approximate some of these parameters by looking at experimental data. And we generally think for kinesin, the real dynamics are somewhere around the orange curve here. Okay, so to kind of say something about these systems, we start by thinking about the entropy production. So stochastic thermodynamics allows us to write down an expression for the entropy production rate for a large collection of systems obeying multipartite dynamics. And so what we find is that the entropy production rate is a sum of terms for each component in the system, all the motors and cargo. And these terms are proportional to the average of the square of this kind of quantity called the local mean velocity, which you can think of as kind of proportional to the probability flux in the system. So we took this expression for the entropy production and applied Jensen's inequality, which basically says that the average of a square of a function is larger than the square of the average of a function. And combining that with the observation that all the components in this system are moving at the same average velocity, we derive what we call the Jensen bound on the entropy production rate for these systems, where we have the total entropy production rate bounded by the square of the mean velocity divided by this quantity called the bare collective diffusivity. And you've got the equation there, but a way to think about this is basically a single parameter that encapsulates contributions from each component of the system. So if you understand how each component of the system, motors and cargo, operates in isolation, then you can calculate this d bare without actually knowing the collective behavior of the system. Those of you who are more familiar with stochastic thermodynamics might notice that this looks an awful lot like the famous thermodynamic uncertainty relation, except with the bare collective diffusivity replaced by the effective diffusivity. And these are 2 distinct bounds. Either one can be tighter in different circumstances. But the important thing here is that the bare collective diffusivity can be calculated just from properties of the individual components of the system. It doesn't require simulating the entire, or measuring the entire system, whereas the effective diffusivity does to actually calculate or measure. So applying this bound to some of the metrics of performance that we want to optimize for these kinds of systems, we can get bounds on many different performance metrics. So in particular, we get an upper bound on the mean velocity of the collective transport system. We get an upper bound on the Stokes efficiency, which is a metric of efficiency that quantifies the ratio of the work the motors do moving the cargo against viscous drag forces to the chemical energy consumed to power them. We also get a lower bound on the coefficient of variation, which kind of quantifies the scale of the fluctuations of how far the cargo is transported in a given amount of time. And crucially, these bounds are totally independent of the interaction potential connecting all the components of these subsystems. They depend only on bare properties of the individual subsystems. Personally, I find the individual performance bounds are more difficult to think about. I like to think about optimizing combinations of two different things. So when motors are identical in this system, and by that I mean they have identical chemical driving forces and bare diffusivities, but potentially generally different couplings between each other and their cargo, we can derive Pareto frontiers that constrain combinations of two different performance metrics. So in particular, we derive a bound on the sum of the Stokes efficiency and mean velocity, as well as a more general relationship between the chemical power consumption and mean velocity of these systems. So going back to this example system introduced earlier, where we have two identical motors and variable barrier heights along their periodic interaction with the substrate, we can look at kind of how these different model curves for performance trade-off curves compare to our theoretical bounds, which are in the black dash lines here. So no real system can beat the black dash line here, where the space is efficiency versus velocity space. And what we find is that as the height of the energy barriers along the motor substrate interaction potential grows, these performance trade-off curves get farther and farther away from the theoretical limit. So the prediction here is that any kind of system that wanted to optimize a combination of these performance metrics would seek to minimize these energy barriers. Although we'll say later whether that actually happens in real systems. And just to say, likewise, we see kind of the same behavior for the other Pareto frontier I showed you, where again, as energy barriers are increased, the system gets farther and farther away from the theoretical limit. Okay, so what about data from real systems? So there's this great paper by Purcell et al. From 2011, where they report experimental data on efficiency and velocity from a variety of different myosin motors in many different animal muscle tissues. Some of these points are like chicken wing and lizard forearm, so really very, very different systems. And kind of taking these data points and plotting them and plugging in our bound with what we think the maximum velocity is roughly for animal myosin velocity, we find that these points are indeed constrained by the Pareto frontier we would predict, but they do not lie along the Pareto frontier. You know, they're a certain distance from the theoretical limit. And that tells us probably that these real systems aren't seeking to optimize just a specific combination of efficiency and velocity. There's at least some other feature at play here. So I want to kind of go back to this idea of interacting subsystems with some kind of black box interaction. And in the last sort of 5 minutes I have here, maybe give you an idea about how we can say something about what's going on in that kind of here be dragons region. Because everything I've told you up until now has been all about constraining the performance of the system if you don't know what's going on in that interaction. And so many molecular machines can be thought of as taking in energy in the form of work from one work reservoir and then outputting work into a different reservoir. The classic example is ATP synthase, which does exactly that. And so generally we would think of them as having some kind of efficiency where they output some work, input some work. The ratio of those is bounded above by 1 thanks to the second law, and so that is a well-defined efficiency. Skipping over a little of the details here, the theory of stochastic thermodynamics allows us to split this single thermodynamic efficiency into contributions from each of the two subsystems, where these contributions kind of relate the free energy going in and out of the subsystems, which I'm not going to get into too much, but it turns out to include contributions from both energy flows and information flows between the two components of the system. These basically quantify free energy inputs and outputs into each subsystem. Combining this framework with the same Jensen bound that we use to constrain the performance of collective motor-driven transport systems, we end up getting upper and lower bounds on each of these two subsystem efficiencies. The derivation is kind of beyond the scope of this talk, but if you're interested, we have a pretty recent preprint going through all the details that I invite you to check out. And so what these bounds allow us to do is to infer the efficiencies of each of the two components in a two-component system entirely from measurable quantities, which basically quantify the interactions of subsystems with their environments. And these are things that can be measured experimentally. So to give you an example, for one of my favorite molecular machines, FO-F1 ATP synthase, which I'll remind you has an FO subunit that has protons flowing across the mitochondrial membrane, spin the FO subunit. It transduces power through basically a crankshaft to the F1 subunit, which also rotates and synthesizes the production of molecules of ATP within the mitochondria. And so, we can write these bounds in terms of quantities that are measurable for this ATP synthase machine. And this allows us to infer what's actually happening inside ATP synthase in terms of the efficiencies of each of the two different components. So we're essentially getting a handle on the free energy that's exchanged between FO and F1 as they're in operation. And these results allow us to fairly tightly constrain the efficiencies of FO and F1. So you can see in the figure there, we've got the efficiency of F1 on the y-axis, the efficiency of FO on the x-axis. Our results suggest that FO is sort of in the range of 50 to 85% efficient, while F1 is more tightly constrained at 70 to 85% efficient. And we can do the same thing for a system consisting of a kinesin motor pulling a diffusive cargo, where there's this great paper from Stradelman in 2008, where they measured a bunch of different things for different cargo diffusivities. And so we can plot those, plug them into our upper and lower bounds on the efficiency of the kinesin motor, and infer kind of a best inferred region in between those upper and lower bounds. And so our results suggest that kinesin is on the order of 70 to 90% efficient. So with that, I'll summarize. So we've got this Jensen bound on the entropy production rate that holds quite generally for multi-component molecular machines, both at the level of subsystem entropy production rates and for the entropy production rate of the system as a whole. This implies new bounds on specific performance metrics of collective transport systems, as well as Pareto frontiers when all the motors are identical, as is the case in many relevant physical systems. If you're interested in reading more, here's the reference. And then likewise, this same tool can be applied to two-component systems to infer subsystem efficiencies in bipartite molecular machines like ATP synthase. And again, here's the reference. So with that, I'll thank all of the different agencies that funded this work. I'll thank everyone in the CIVAC group for helpful discussions, and especially my supervisor, David. And then I also want to acknowledge that all of this work was done on the traditional unceded territory of the Squamish, Tsleil-Waututh, and Musqueam First Nations around Vancouver, Canada. So with that, I'll take any questions. Yes. Yeah.
I can see it from that.
Yeah. Yeah, no, that is kind of part of the point, is that it didn't come back because we don't need it. So the results are independent. Like, this potential can be anything subject to the constraint is, I didn't say what the constraint on the potential is, but it has to be such that all the components of the system stay together at long times. So they can't become infinitely separated as time goes to infinity. That's basically, mathematically, that's the constraint on the potential. But other than that, it can take any kind of form. So like, there might be long-range cooperative interactions between motors mediated through the microtubule. There's some experimental evidence for that. There might be short-range repulsive forces. There could be linkers between the motor and cargo with a preferred rest length. Like, really, you can put anything into this framework, and the results follow. Yes. Two sequential processes. Can you elaborate on what you mean by two sequential processes? Oh, okay, I see what you mean. I'm pretty sure our results would still apply in that case. Like, we're not assuming we can measure the positions of independent motors or anything like that. The only result that really, or the quantity of the collective system as a whole that would be measured that really comes into this is the average velocity of the system. So I think that's typically measured by people just measuring the position of the cargo as it moves along, because that's kind of the bigger object, easier to see experimentally. Yes.
So if I understand correctly, what you've done is to say that the
Right, yes.
So again, I guess I would have been more comfortable if there was another coordinate, somehow the chemical coordinate, you push on that, probably follow.
Right.
Somehow, so you must be in some adiabatic limit where that goes away.
Not so much an adiabatic limit. It's more it's really the tight coupling limit. So we're assuming that there can't be, like, if we think about like the chemical mechanical space as a 2 degrees freedom, 2 degrees of freedom, we're assuming that any motion in the direction of the chemical coordinate automatically leads to motion in the mechanical coordinate. So there's no freedom there. And that is consistent with what's found experimentally for kinesin, at least. Again, I'm less certain about other kinds of motors, and we're actively working on relaxing that assumption. So that's something I hope that we'll address in the coming months, because it is for sure an interesting question of what if you had these orthogonal mechanical and chemical degrees of freedom. So I have a pretty strong hunch that our results still hold in that case, but we haven't proven it with 100% certainty. So yeah. Hopefully that'll be coming up.
Thank you. Thank you to the organizers for having me. So I'm excited to be here to present this work. This started as a student project during the 2019 Boulder Summer School, and I kept working on it with Caroline, among others, and it's about irreversibility in flocking. Sorry. I want to start with a video. It just lasts for a few seconds. And I would like to ask you at the end of it, if you think that this is played forward or backward in time. Okay, it's almost done. Okay, that's it. How many of you think this is forward? Okay, and how many of you that this is backward? Okay, there is an imbalance, surprisingly, towards forward. That's actually true. Does anybody have a reason for that? Yeah? Oh, I'm pretty sure I concealed that, but okay. Oh, okay. That's somehow. I should shade more. Okay. Well, the point I wanted to make is that it's actually hard to tell that just by looking at the birds, especially at that distance when we don't have access to, well, first of all, we cannot distinguish between tail and head. And this is somehow related to the way we interpret the various degrees of freedom and their parity under time reversal. And also we don't have access to the biological details that allow them to move, to keep this state of motion. And that is done, of course, through highly reversible processes. So what I want to do is, first of all, identify signatures of irreversibility in these models, and to tell what are the equilibrium limits, if there exist equilibrium limits, and when they do occur. And finally, another question that is of interest is there anything that we can learn from irreversibility about the interaction structure in these systems? So we are in this very nice intersection of three different subjects. There are interacting complex systems, active matter, and non-equilibrium thermodynamics. And we want to use the tools from stochastic thermodynamics, I'm very glad you introduced that, to answer these questions. Now, the importance of irreversibility for this model has been stressed since the beginning, since the introduction of the VHSEC model, because it was very clear that the complex emerging phenomena were a manifestation of their non-equilibrium nature. In all the models that have been introduced to describe, to give a minimal description of what is called polar dilute dry active matter, contain these three fundamental ingredients: directional degrees of freedom, ferromagnetic short-range interactions through which these directional degrees of freedom are aligned, and self-propulsion. And it is through self-propulsion that we can actually have true long-range polar order, even in 2 dimensions, a fact which is not possible at equilibrium in the absence of self-propulsion because of the Mermin-Wagner theorem. However, it has been recently shown that if one takes a system of self-propelled motile particles, but enforces the dynamics to respect detailed balance, then there cannot be any spontaneous symmetry breaking of a continuous rotational symmetry in 2 dimensions. So we have a generalized Hohenberg-Mermin-Wagner type theorem. So this means that it is not self-propulsion per se, the crucial ingredient, but the fact that self-propulsion breaks detailed balance, and so irreversibility is key. So what we want to do is to measure irreversibility in some minimal flocking model. And the first 2 questions that we have to answer when we set up the problem is, at which scale are we interested in describing the system? And as I anticipated, it's the macroscopic scale. The same questions, however, can also be addressed at the level of continuous field theories. And the second question is, what do we want to measure? There are many quantities that can be used for this purpose. And one of them is, for instance, effective temperatures, measuring the violation of fluctuation dissipation theorems, or we can use currents in the phase space of the system, or we can use the tools from stochastic thermodynamics, so energetic fluxes and entropy production in particular. And we will focus on entropy production for this model, that is the active XY model. So we're working in 2 dimensions, and the dynamics of the system, we have a continuous rotational symmetry in 2 dimensions. The dynamics is described by this set of differential equations. The first one just tells us that the particle's position follow deterministically the velocities, and the velocity orientations, all the velocity vectors have a fixed modulus V0, and the velocity orientations evolve in time according to this Langevin process that just describes the equilibrium dynamics of an XY model. I mean, it would describe the equilibrium dynamics of an XY model if the Nij matrix was symmetric and derived, for instance, from the lattice structure. But in these systems, the interaction matrix is determined after the positions of the particles. And since particles move, are motile, this is time dependent. So we can interpret this time dependency as an effective protocol that drives the system out of equilibrium. It is not an external protocol in the sense that it is not fully independent from the dynamics of the theta variables. And it is this coupling, as we will see, that creates the irreversibility and dissipation in the system. So we want to measure entropy production. We'll recall the definition. It's just the Kullback-Leibler divergence between forward and backward probabilities of forward and backward trajectories. And in particular, we're interested in the entropy production rate, which is defined as the intrinsic quantity in the length of the trajectories, in the limit of infinitely long trajectories. We also need to specify the parity under time reversal of the state variable coordinates, and we assume that positions are even, whereas velocities are odd. So the theta variables are mapped into theta plus pi. But in active matter, this is a controversial issue. However, with this interpretation, we're able to find 2 equivalent expressions for the entropy production rate. The first one can be interpreted as the rate of stochastic heat that this system of spins exchange with the heat bath that is at temperature T. And the second expression can be interpreted as the work of some fictitious reshuffling forces that rewire the interaction network. And these 2 are equivalent if we are in a steady state, because they just differ for a state function that is the average of the average energy of the XY model. We check that we are effectively in this condition from numerical simulations. Having these expressions, we could measure them from numerical simulations. We consider 2 variants of the model. The first one implements metric interactions. We assume that each particle interacts with the other particles that are within a distance equal to r. And in the second case, we are implementing topological interactions, assuming that each bird only interacts with the first shell of Voronoi neighbors. And it is known that these 2 models, these 2 variants of the model have different phenomenology, in particular, the presence of a phase coexistence associated to a first order phase transition in the first case, whereas in the second case, the phenomenology is compatible with a second order like transition. And you can also see that from, in the first case, from the distribution of the number of neighbors signaling this coexistence of dilute and dense phases. And correspondingly, we have different shapes for the entropy production rate curves. Here it's the entropy production rate per particle divided by the size of the system. It's also in different, I mean, I'm also using different scales, so they're not as different as they seem here, but the major difference regards this region where we have a kind of plateau for the metric model that also corresponds to the coexistence region. Whereas in the second case, we don't have this kind of plateau, and the collapse of the different curves is much better for all the values of the control parameter D. In particular, we also see that we have a good agreement with the scaling of the entropy production rate in the limit of small d, that is small temperature or high temperature. We have indeed two equilibrium limits in these two regimes, the deeply ordered regime as d goes to 0, and the strongly disordered regime as d goes to infinity. And the peak corresponds to the transition point. So this is easy to explain if we recall the second expression for the entropy production rate, because this was interpreted as the work of reshuffling forces, and reshuffling is maximal at the transition point. On the contrary, if we are in the deeply ordered regime, reshuffling is suppressed by the presence of polar order because all the birds move in the same direction. There is just a little bit of transverse diffusion. And this suppression is faster than D. I mean, this is what the data is telling us because we have also a factor 1 over D in the expression. In the high temperature limit, D equal 5 in this case, what happens is that since D is at the same time the temperature of the heat bath that the spins are in contact with, but it can also be interpreted as the rotational diffusion coefficient of the active Brownian particle. Having high D means that the particles quickly reorient themselves without moving away from their original position. So they don't exchange the neighbors very efficiently. And this is what suppresses reshuffling. On the contrary, close to the transition, we have a motion that is persistent enough, and we're fully exploiting this persistence. We're not subtracting a collective direction of motion because we are just at the onset of collective motion. And the same thing can be also observed from the autocorrelation function of the network that decays the fastest, correspondingly to the red curve. I'm not sure it's very clear, but it's the fastest decay close to the transition point. So, okay, we have a mechanistic understanding. This is not new. It was already clear that reshuffling was at the basis of dissipation, of irreversibility in these systems, but it had not been quantified across the phase diagram. What can we tell more about that, having this interpretation? We can tell something more in the case of metric interactions, because here the expression for the connectivity metrics is simple enough that we can easily compute the time derivative and plug it into our formula. So since reshuffling occurs when a bird crosses the boundary of the interaction disk, we can basically focus on just pairs of particles that are at a distance equal to r. And to characterize the configuration of this pair of particles in 2 dimensions, we just need 2 angles. They're alpha and phi, defined as in this picture. So phi is just the angular difference between the orientations of the 2 birds, and alpha is the angular orientation of the displacement vector with respect to the collective direction of motion. I mean, not collective at the collective level, just for the 2 particles. And this means that we can rewrite the expression for the entropy production rate as an average over the distribution of pairs of particles of this entropy production rate per pair that has this functional form plotted here, which obviously is completely anti-symmetric under horizontal shifts of pi, because this is the action of the time reversal operator. So in order to have a positive entropy production rate, a non-zero entropy production rate, we cannot have mirror configurations like the black arrows and the pink arrows that are equally probable. They cannot have the same probability if we want to have positive entropy production rate. So this asymmetry is a local signature of irreversibility that occurs at the scale of the interaction radius, and that we could check in our numerical simulations. Here I'm plotting the logarithm of the distribution of pairs of particles that exhibits this little asymmetry that is positively correlated with the anti-symmetry of epsilon. And this is even more evident, this positive correlation is even more evident if I'm just plotting the anti-symmetric part of this log distribution. Another thing that we could check is how this asymmetry varies as we vary the distance between the particles. And we can see a clear peak at a distance equal to the interaction radius for this interaction kernel. So this suggests that this asymmetry is also encoding some information about the interaction kernel, the structure of the interaction kernel, and that is something that can possibly be used to infer something from real data. I want to conclude, just saying that these local signatures of irreversibility by means of pair asymmetries is just a manifestation of a more general result that is not just valid for the active XY model, but for a broader class of stochastic systems that we dubbed irreversibility-induced explicit symmetry break-in. And basically in the active XY model, what this consists of is that the condition of irreversibility implies an explicit breakdown of the symmetry under rotations in the subspace of velocities, which is what is typically considered as the starting point for Mermin-Wagner theorems. This is not just possible for active XY model. As I was saying, it's a more general result, and we have something similar. I don't want to go through the details, but a similar asymmetry can be derived from the condition of irreversibility for any stochastic process that has a zero reversible drift and additive noise. So to conclude, what we understood is that, as it was already known, irreversibility comes from the coupling between self-repulsion and interaction, and it can be rationalized through reshuffling. This tells us why we were not able to distinguish between forward and backward directions in the original movie, because birds are in a condition of local equilibrium, as it was already put forward in 2016, this paper, where they measured from real data the timescales related to the relaxation of the velocity correlations and at the scale of the interaction radius, and the neighbor exchange time tau network that corresponds to the red data. And there is a clear separation of scales, telling us that the real bird flocks operate in a condition of local equilibrium. On the contrary, there are other animal systems like insect swarms that seem to be close to critical point. They're not ordered, so they're probably in a condition similar to the one where reshuffling is maximal. And it has been indeed observed again from the data that the reshuffling times and the decay of the correlation function, the timescales of the decay of the correlation function are comparable irrespectively of how one defines the neighborhood. So these are candidate systems to observe these asymmetries. The advantage of having a model that tells us that these local signatures of irreversibility come in the form of pair asymmetries is that we are reducing the dimensionality of the things we want to observe, and this becomes statistically accessible with the data that we have at the moment. So yes, the hope is to apply this to real systems and possibly to also use this asymmetry to learn something more or more detailed about the interaction kernel in these systems. And with this, I conclude. I want to thank, of course, my collaborators, Caroline, Jordan, and Simon from the Boulder Summer School, and my supervisors and the institutions that hosted me while I was working on that. Thank you. Oh, you mean, well, how do you know that over long times it doesn't coalesce into a single blob? Well, we ran very long simulations and we didn't observe that. We are also, what prevents this from happening here is the noise. So yes, this model is a bit pathological, and the density that you reach at small temperature is very high. So we don't have repulsion. It's just alignment interactions, and it is known, and interactions are such that we are, where is the Hamiltonian? Okay, here is the Hamiltonian. As you see, in contrast to the original Vichsek model, we're not dividing by the number of neighbors in the disk, which creates a very strong feedback between alignment and density fluctuations, which causes this first order phenomenology and very strong phase coexistence. The intermediate temperatures, what prevents this from happening, at least on very long time scales, that are the time scales we observed, we don't have coalescence into a single one because there is noise.
Go back one slide to the, yeah, so you break down the entry production of these two parameters, this E and the shuffling board.
No, it's not breakdown. There are two equivalent formulas. So basically, you can see them going from S1 to S2 by simple integration by parts if you want. And yeah, I mean, equivalent up to a boundary term that we assume, I mean, we check that from simulations, but assuming that this is a state function or that it changes sublinearly with time, we can tell that for the entropy reduction rate, these two are equivalent. Yeah, yeah, because well, like in MIPS, motility induced, so scalar active matter, where you just have active Brownian particles with repulsive and attractive, just repulsive is enough to have the phenomenology. With repulsive interactions and no alignment, they interpret this term that is here as a self-repulsive force that doesn't change sign on time reversal, but they add here some noise. So it's not completely, it would be completely irreversible in that case. Well, the entropy production rate would diverge. But if they add some translational noise here that we don't have, we just have rotational noise, then they can apply the same formalism, and this is how they typically compute entropy production rate in MIPS. They interpret. Yeah, like you, in that case, you have access to the tail. I would say you have access to the tail and head position, and you play it's like the way you did for this movie. It's completely reversible if you don't have noise because of that.
Not only has the direction, but also error embedded. I can tell whether I'm moving in the direction of the error.
Yeah, exactly. Exactly. But I mean, it's an open question in what is the best prescription in those models where they have an interpretation for that as a force, and they interpret this first equation here as an overdamped equation. So here reshuffling is that birds change their neighbors in a static. Yeah, we have an Nij. Sorry, I didn't specify that, but the Nij matrix here it takes values 1 or 0 depending on whether bird I and j interact with each other or not. And this is not a constant property of the pair. It changes over time because the Nij matrix is determined after. I mean, with these two rules, respectively, but since particles are motile, they change in time. So yeah, it's spiking. If we take this formalism, we have like spikes, deltas, but we're taking an average. So we're assuming some ergodicity here to go from the limit over tau of the average stochastic entropy divided by the length of the trajectory to the average over the steady state distribution. And we're assuming we're in a proper non-equilibrium steady state because of this numerical check that this function is not changing.
So is this statement the next page? Yeah, if your couplings are stronger than
It's not the couplings. So we did not vary the strength of the couplings here. We just varied two control parameters. I'm just showing one here that is this D parameter that corresponds to the temperature in this spin model, or if you prefer, the rotational diffusion coefficient of the active Brownian particle. They can be interpreted in both ways. And is this very large, meaning high temperature, we're in a disordered state. But since it's also interpreted as the rotational diffusion coefficient, the particles just reorient themselves. They don't move in a persistent way.
sort of like
Yeah, of course. But it's a noise that acts on the orientational dynamics. Yeah, in that case, the point is that you can think of the system, the reference system, as an equilibrium ferromagnet with a fixed freezed configuration that moves in an inertial frame that is co-moving with the center of mass of the flock. So but this is only possible if you're interpreting the velocities with the right parity, and that's it. And it was not obvious to us at the beginning that this should be 0, even if there is this equilibrium limit, because we also have a factor 1/d here. So yeah, reshuffling is.
Yeah, it's small. But I guess so. You're saying in the frame, I mean, there's
It's almost equilibrium, yeah.
Yeah, is that like you can put it back going down? But
Yeah, but then you don't have a preferred direction.
Yeah, no, that's.
Well, after having these curves, we worked a simple dimensional argument to understand the scaling in the two limits, and it's compatible with that. So one can also understand that with simple dimensional arguments, analyzing the magnitude of the terms that appear in this expression here.
Very quickly, you said that in the case of the metric interaction, first order transition, it sounds like the thing when did this settle?
So yeah, where is Luca? Yeah, it is quite settled. So it's settled for the metric case. It's clear that it is first order from simulations, from linear stability analysis. It's not a definitive answer, but one simple way to see that is from linear stability analysis that shows that in the field theory that the uniform state, homogeneous state, polar state is not stable. Whereas for the topological case, it's under debate. Also, there are different ways of implementing the topological interaction. In this case, I want to stress that we always had symmetric interactions. Because if you add non-reciprocal interactions, this is, of course, the second source of irreversibility in the system. We worked with the Voronoi tessellation for the topological case because it enforced this. If you take like the first n nearest neighbors, you can have asymmetries, and this can lead to a slightly different phenomenology for what we were interested in. But it is also something, I think, for the K and N model, that is what I was describing, the first K nearest neighbors. People have run extensive simulations very long simulations and very, very large sizes, and saw some bands in that case. Bands at close to the transition point. So like Julien Tailleur and friends are pushing this. But I don't know, I think this is still open and it can depend a lot on the microscopic details. Sorry? No, there is, it was just simple to implement, and but one can also, I actually also ran simulations with that decay. Actually, no, I work with an exponential decay. So I think the difference is when you go from short range to long range, but as long as you work with short range interactions, shouldn't change much, right?
And if you have a weighted, continuous weighted, presumably Nij will replace the part, WIJ, and everything will come out the same way.
Yeah, no, this is, I think, what it was. I think it was basically what he was asking. I replaced the definition of Nij. Oh, the formula doesn't change. It doesn't change. We're not assuming that Nij is discrete here. We're just using this in the simulation.
Yes, so thank you. Good afternoon, everyone. Before beginning, I want to thank the Center for the Physics of the Biological Function for inviting me and to give me the chance to present to you our work. So in this talk, I will present to you an experimental approach that we introduced in the lab that, if combined with mathematical modeling, has proved to be effective in studying the sensory systems. So when we're faced with a sensory system, a typical way, classical way of studying it is present to it some stimuli and record responses from it. And this is a very general paradigm, and in particular, the sensory system that we are interested in is the retina. And so for us, stimuli are 2D images and responses are spike trains that are recorded from sensory neurons that compose the retina. But beyond the retina, we can call this stimuli just X, whatever they may be, without loss of generality, and the response is just Y. And so the sensory system becomes a function F that takes the stimulus X and transforms it into Y. So when studying sensory system, for us, it's crucial to understand what are the features of X that drive the response Y. And so in other terms, what are the changes of X that are relevant for the function F? Or if you want, conversely, what are the changes of X that leave F invariant? And this is the big question that we want to address in this talk. And so I want to motivate to you why this is interesting, and I want to do it with a little illusion, visual illusion. So if you take this bunch of ellipses, you will notice that they are of the same color and they are superimposed on some striped background. So what I'm going to do now is I'm going to keep this image and just change the context in which you perceive those ellipses by adding just lines on top of them. So, and you will notice that even though I swear to you that these and these are the same ellipses, depending on which lines I decide to enhance from the background, you will perceive the ellipses in different color. And this is because not all the information that is present in the image X in terms of pixel intensities arrives to our perception. The perception is shaped by the context in which you perceive X. And so when studying perception, we want to understand, and it's interesting to understand, what is the part of the information in X that is retained by perception and what part of the information is discarded. And so people tried to do this in the past, and they tried to do it by approximating linearly the function F. And so writing the response Y just as a linear projection of the stimulus X on some linear filter that here I call H. And this has the advantages that it's simple and it's also easy to interpret, because if we imagine a high dimensional stimulus space and we imagine H as a direction in this stimulus, then X can always be decomposed in components parallel and orthogonal to H. And by construction, the changes that are going to be relevant for F in X are the ones that are parallel to H. And so to address the question of what are the relevant changes in X, we just have to measure H and we are done. And luckily, it's easy to find an optimal solution for H that we will call from now on receptive field, and you will understand in a second why. And this solution takes the form of basically a weighted average of the stimulus by the response, normalized by the autocorrelation function of the stimulus. And the way people set out to measure this H was through stimuli that are called white noise checkerboards that look like this. And they use them because by construction, they have a stimulus autocorrelation function that is identity because they're noisy. And so here, the receptive field is just the weighted average of the stimulus by the response. And that's exactly how they measured it. So they showed a sequence of these noisy frames and they recorded the response of the sensory neuron to those frames. And then they could just do a weighted average of the frames by the number of spikes they evoked. And this procedure is called spike triggered average, or STA for short. And it can give two outputs, one of two outputs, either a so-called on STA, so a positive polarity 2D Gaussian, or an off STA, a negative polarity 2D Gaussian. And this STA gives us two types of information. One is from the position of the 2D Gaussian, and the position tells us where the position compared to the image that we're showing, and it tells us where is what part of the visual field our sensory neuron is affected by, and are these pixels that are inside the 2D ellipse. And then the polarity of the field tells us whether this neuron is encoding, so it's increasing its response when we increase the light inside its receptive field, so it increases the luminance, we say, or when we decrease the luminance. So for the on, the neuron will increase the firing at an increase of luminance, while for off, it will increase the firing for a decrease of luminance. And once we have this H, we can test the accuracy of this linear approximation by using this filter H as a model. So we take an input stimulus, we project it on the filter H, and then we just do add a nonlinearity that is a positive threshold that just prevents this model from predicting negative number of spikes, because this is against biological constraints. And so we can use this simple model that people call LN, because it's composed of a linear and nonlinear part. And we can use it to predict, as here in red, the responses to the stimulus, to the input stimulus, and then we can confront this with the black actual response of the retina to the stimulus. And as you can see, for this white noise stimulus, this linear nonlinear model does pretty well. But the problem is that for natural scenes that are taken from the natural world that are more complex, like this one, this guy with the umbrella, this linear model fails pretty badly. It still captures something, but it's not behaving. And this is because the complex structure of this stimuli drives higher nonlinearities in the F, in the function of the sensory system. And so this linear approximation or semi-linear approximation is incorrect. So we were interested in studying these natural scenes, and with our lab just took a step back to the question. We wanted, as I said, to characterize what is relevant inside of the X for F, of the input X for the function F. And so we said, well, even if F is highly nonlinear, we are sure that at least locally around some stimulus X0, the selectivity, so what is irrelevant for the function F, will be dictated by the gradient of F calculated in this X0. So yes. Yeah. Usually no, it's obtained with the white noise, and this is because Yes. Yes. Yeah. Yes. Yes. If this H was a property of the neuron that generalized to any stimulus, then this shouldn't matter. You can do it you can estimate H on the natural images. You can do this weighted average, but first of all, it shouldn't change if the linear approximation was correct. And second of all, if you did that, you would have to invert the stimulus autocorrelation matrix, which is really large because these are really high dimensional stimuli, and so it becomes numerically hard. So we want to characterize the function f like this, but as much as we know what x0 is, which is a particular natural image, and what f0 of f of x0 is, which is the response of the sensory neuron to this image, we don't know what we don't know what f prime of x0 is and how to measure it. And also, we want to see if we can, by measuring f prime of x0 in a lot of x0s across the stimulus space, we can characterize what visual feature this sensory neuron is responding to. And so we propose to measure this f prime of x0 with an approach that we call perturbative. So we propose to add on top of the reference image x0 some perturbation, white perturbation delta, that will give us an image x that looks like that. And delta needs to be noisy, so we need to have an average mean at 0, and it also needs to be small. Small in the sense that the response can be approximated as to be linear in the perturbation. And once we have calibrated this checkerboard pattern for this to be true, we can, instead of showing the reference image, show it over 1,000 times, repeating it, but each time superimposing it with a different perturbation. And then we can record the response of the neuron to this, and we can again do a weighted average of the perturbation, this time by the response. And much like in the STA case, we obtain another filter that we call local STA. That is not the receptive field of the neuron overall, but it's an approximate estimation of the local receptive field of the neuron around the reference image x0. And if we write out this weighted average that we call the local STA here in the and we assume the linearization of the response to be true, then we can simply see that this local STA is proportional to the gradient f prime of x0. So that's the observable we wanted. And I can also give you another interpretation of local STA. So this would be the change in this that we should apply to the stimulus x0 in order to make the response of the sensory neuron to maximally increase. So if we see the natural image as a point in a highly dimensional stimulus space, then the local STA is the direction in which we have to move in this stimulus space in order for the neuron to maximally increase its response. So the local STAs, like the STAs, can be on or off. And again, if they have information encoding interpretation in the sense that when a neuron has an on local STA, it means that for this, around this image, it will be selective responding to light increases, while if it has an off local STA, it will be responding to light decreases. So now, if we imagine, if we measure these local STAs for a lot of x0, so a lot of natural images across the stimulus space, and we imagine that this sensory neuron is encoding for luminance, for example, so for luminance increases, for example, across the stimulus space, then we would expect to be in a situation like this one, where no matter around what point you measure the gradient, you will always have the gradient to be parallel, either on or off, for example. And this is what the linear approximation would predict also. And these neurons, there are a lot of them, and we call them luminance encoding cells, because by reading the response of these neurons, you can reliably encode for luminance, because each time you see them increasing their firing rate, it means that the luminance in the receptive field is increasing. And as I said, this is the case for many cells, but in around 20% of the cases, we actually see something like this. So depending where we measure the gradient in the stimulus space around which image, the on-off polarity of these local STAs, we call it polarity, can change. So we showed that the on-off selectivity changes with the natural stimuli. And so as I said before, this is it's beyond the linear approximation. So the linear model doesn't work anymore in this case, and we want to see if we can find a model that actually reproduces this on-off selectivity change. And we also want to ask ourselves, if we see this change, then it means that the neuron cannot be encoding for luminance, because by reading an increase in its response, we will not know if it's for this image and a luminance increase or this image and luminance decrease, for example. So the signal would be ambiguous. So probably this neuron is encoding for another visual feature that is not luminance. And so can we, by sampling the stimulus space, can we retrieve that? So we start by looking for a model that reproduces this polarity inversion. And to do that, we present the retina with 3,000 unperturbed natural images, so just the images. And we trained 2 models to reproduce the retinal response to them. And then we can derive these 2 models, differentiate these 2 models, calculate their gradient around the natural images, and obtain a model prediction of the local STAs. And we train a simple LN model like the one I proposed to you before, described to you before as a benchmark, and a CNN, a convolutional neural network that, despite its scary name, it's actually a pretty simple model, and I will describe it to you in 2 seconds. And then we can confront the model predicted local STAs with the local STAs that we measured with the perturbations. And we see, as expected, that the LN cannot capture this polarity change from on to off, while the CNN can. And I want to give you an intuition of why the CNN can do that. It's because the CNN has a first convolutional layer that is composed of two channels that, once they're trained, they look like this. So they are an off and an on channel. And the input image is processed parallelly by these 2 channels. And then the output of these channels is passed through a nonlinearity, and then the 2 pieces of information are combined again inside the sensory neuron we are recording from. And so it can happen one of 2 things. When we present an image, it can happen that it has a high activation on the on channel and the off channel, and so it passes the off nonlinearity, but it doesn't pass the on nonlinearity, and then the model will predict an off local STA. While conversely, if the image activates strongly the on channel, it will pass the on nonlinearity but not the off, and the model will produce an on local STA, will predict an on local STA. So it has a deep net name, but actually the fundamental part of this model is just that there is a parallel on-off processing of the information that then we pull together. So we showed that selectivity to light increases or decreases is context dependent. And we showed that this can be reproduced by a nonlinear 2-layer convolutional model. We still have to investigate what does this mean in terms of what the neuron is responding to. And to do that, we use the model to predict the local STAs for all the 3,000 images. And we have a set of 3,000 local STAs. We can take that and do a principal component analysis on it, and we end up for every cell with 2 principal components that look like these 2, and they account on average for 90% of the variance in the local STA dataset. And we can use those 2 principal components as axes on a plane, and we can project on these planes, displaying all the natural image local STA couples that we predicted with the model. And we can, for example, take this image here and project it on the 2 components to obtain this point. And then we can take the image plus the local STA and project it again to obtain this other point here, and then link the 2 with an arrow. And if we repeat this for the 3,000 natural images, for those cells that show this on-off polarity change across local STAs, we see a vector field that has a structure like this one. So it's not a random variation, but it's has a structure in the stimulus space. And this type of vector field is quadratic, and it has a pre-intuitive explanation, and I will try to give you an intuition of. So this point actually corresponds to a gray screen. It's the projection of a gray screen. And so the arrows that fall on this right side of the graph are arrows that begin around a dark image inside the receptive field of the ganglion of the sensory neuron, while the images that are here on the left are images that are bright inside the receptive field of the neuron, and the local STAs vary accordingly. So if the neuron is presented with a dark image, the local STA will be off. So it will be the neuron will want you to apply a perturbation to the stimulus that makes the image inside its receptive field even darker. While if you probe it with a bright image, it will want a perturbation that makes this image even brighter. So bright gets brighter and dark gets darker. And what this neuron is actually selective to, so when this neuron is increasing its firing rate every time that I'm taking it as far as possible from the gray. So the neuron is trying to maximize the contrast inside its receptive field. And to benchmark that, we came up with a simple contrast model in which the response of the neuron is modeled by just a weighted sum of the square difference of the pixel values of the image to some gray level. And if we use this simple model to predict we train it to predict the retinal response, then we use this to predict the local STAs, and we project the local STAs again on these two principal components, we obtain a vector field that looks like the one here in green. And so, as you see, the contrast model is not capturing the whole variance of the CNN, but it does reproduce this quadratic shape vector field. And also we went back to the data and we looked at natural images and neurons in which, and so happened that an edge of the natural image fell inside the receptive field of the neuron. And you see that the local STAs are the ones that make this edge even sharper. So make the dark parts get darker and the bright part get brighter so that the edge gets sharper and the contrast content inside the receptive field is maximized. So to conclude, we showed that the selectivity to light increases or decreases is highly context dependent for natural scenes, that this dependency can be reproduced by a nonlinear two-layer convolutional model, and that this dependency suggests a robust coding of contrast. So on our part, what we want to do in the future is to extend this perturbative approach to time, which we integrated out in this analysis, but we want to take that into account and use more dynamical stimulation. And also we believe that this perturbative approach could be used in other sensory modalities to characterize other sensory systems. And with that, I want to thank you and thank all the members of my lab, and particularly Matthias Golding and Baptiste Lefebvre that are co-first authors with me on this work, and my supervisor, Olivier Mar. And by the way, we did publish this work. I forgot to put the reference here. And also Olivier is recruiting. So if you like what we do or you're interested, at least you can send him an email at this email here and get in contact. Thank you. Yeah. It can be, yes. Color processing is something surprisingly little studied in the retina, but we could do that, yes. It's just a matter of, for us, it was just a matter of like even a technical question, like we didn't have color stimulation, but it can be done for sure. We're actually thinking about doing two types of perturbations in two different colors, like showing colorful images and doing perturbations in different colors. and see how if this changes and how this changes across stimulus space, but we haven't done that. It's one of the directions. Yes. How they are done? The experiments, well, basically what we do here, I didn't say it, but well, first of all, you have to dissect the retina from the eye. So you take the retina from the back of the eye of some animal, for example, mouse. We did this in mouse and salamander, and you take it, you dissect it, you place it on a multi-electrode array, and you project on the retina, on the photoreceptors level, your image, your stimulus, and then you record the response of the output neurons of the retina, because the retina is organized in layers of neurons, and there is the last layer of neurons is the ganglion cell layer, and these are the neurons that with their axon compose the optic nerve that goes directly to the brain, and we record from there. In terms of stimulation, when I said that we integrate out the time, it's because we flash these reference images for 300 milliseconds. We count the number of spikes that they evoked in these 300 milliseconds, and then we decorrelate the stimulus with a gray background, and then we do another image for 300 milliseconds and so on. So now we're thinking about going to something more dynamical. Oh, Pi response. Oh, there is a question in chat. Is this model strong against adversarial or examples attacks? This is a good question, and the true answer is that I don't know. We didn't check for that. I don't know. It's a good question. I don't know, because we're not really a machine learning lab. This thing is called a CNN, but it can also be called a LN model, like a two-layer linear nonlinear cascade. It's the fact that is DeepNet is just because we wanted to fit this first convolutional layer not at the single cell level but on the neuronal population level. So we designed a convolutional layer structure, but we didn't investigate it any further on the machine learning side.
So do I understand correctly that at the end, the response is some complicated nonlinear function?
Yes. It's because, I think it's because this depends on the characteristic of the stimulus. Basically, we use the stimulus that, okay, it's complex spatially, but in the end, it's fairly simple in terms of it's matched in contrast, it's matched in luminance across various frames, and it's just
I mean, there are, if you knew that what you were searching for, then you can sort of.
yes. Well, yeah. Yes, that's true. But we didn't know. In fact, we figured it out as we were going. But yes. I think there is another one. Thanks. That's a good question. Yes, I don't know. I think you could probably reconstruct edges, but yeah, I don't know. Decoding, it's always a very, very hard problem, and I think so. I have an hunch for it, but we haven't tried anything in this sense. So
when you show these the weights of it, you go to one of the pictures where you show like the weights of different
like that.
Yeah.
Yeah.
The sort of the way that the response of the neuron depends on the larger yet the response is also going to be pretty localized. Where do you think this information about context is how the cell generated information from like other cells higher up about the rest of the
Yes. Actually, we have a pharmacology part in the paper that I didn't mention given the audience, but in the retina there are some cells that are higher up in the hierarchy before ganglion cells, and they're called bipolar cells. And actually, they are distinguished in two types. There are on bipolar cells and off bipolar cells. So the on bipolar cells are activated by light increases, and the off bipolar cells are activated by light decreases. And this behavior of these ganglion cells is actually for the ganglion cells to behave like this, it has to pull information from both these two pathways. And we actually prove that by blocking one of the two pathways with pharmacology, and we see that if we block the on pathway, for example, the on local STAs disappear in the perturbation without any modeling. They just disappear in the data. Yes. 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 It's correct. Yes. You can. Actually, we did the stimulus in a way that the adaptation shouldn't matter, because at least the contrast and luminance adaptation, it didn't matter because we matched the single frames of the stimulus to have the same contrast and same luminance, at least globally. We couldn't match them inside the receptive field because we didn't know a priori where the receptive field was, but across the image we did. And you can tell them apart in the sense that the contrast I've been talking about is spatial and it's to some gray thing, while the adaptation, it's more a temporal phenomenon that happens in time. And actually, one way that I would like to expand this project into would be to, instead of going full in the time component and just present a dynamical stimulus, I would be interested to redo this perturbation measured of the local STAs after I condition the retina to adapt to some different feature of the stimulus. And I want to see if we see the same thing or how does it change. And that's a great question. I really think this is the game in the future.